Morning, everyone.
Good morning.
Good morning.
Thank you. Good morning. Welcome back. Thanks for getting settled in. And of course, to our virtual audience, good morning, good afternoon, and good evening, wherever you're joining from today. Welcome back to the Infant Nutrition Science Coalition's workshop. Again, my name is Cypress Links. I'm a scientific program manager at LCUS in Canada. If you are joining us for the first time today, I just wanted to go over a few housekeeping notes. So the workshop is being recorded, and the videos, including the previous sessions, if you missed them yesterday, will be posted to our website in the coming weeks. Throughout the workshop today, we're going to present questions to the audience for their input through Slido, as you can see on the slide here. And you can scan this QR code to enter the Slido discussion. It'll also be displayed as we ask each question. And you can also go to slido.com and use the code 4190806 to enter in your responses when we have those discussion portions. Both in-person and virtual audience members are encouraged to respond on Slido. It helps us collect your input as well. And for our virtual audience, please don't hesitate to use that Q&A box and go to webinar to add any context to your responses for Slido. We're capturing all of that for the workshop outcomes. So during those discussion portions, for those in the room, as a reminder, please use the microphones on your table. Remember, you've got to flip the little switch to turn them on. And then please also identify yourself before you speak, just so everyone in the room and online knows who's speaking. And then if you're joining us virtually, you can also use that Q&A box to enter in your questions, and we will ask them to the moderator during the discussion portions. Please be aware that due to a tight schedule today, we may not be able to get to everybody's question. So that is all the housekeeping I have for you. I'd now like to welcome to the stage two of our colleagues from Oregon State University, who are both authors on the prospectus paper that we described yesterday. They've been taking notes throughout the workshop, and we're going to provide us with a brief summary from the first two sessions to remind us of key takeaways as we prepare for our final session. So I would like to welcome to the stage Marie Ryan, who's a research assistant at Oregon State University with the Dallas Lab, and Clay Swackhammer, who's a research associate affiliated with the Dallas Lab and Moore Family Center for Whole Grain Foods, Nutrition, and Preventative Health at Oregon State University.
And I'm filling in for Clay today, so stay Dallas.
Okay, good morning, and thank you for attending day two of the Infant Nutrition Science Coalition workshop. I am Marie Ryan, a researcher with Oregon State University and major contributor to the Perspectives paper that was pre-workshop. This morning, I'm going to give a brief recap of yesterday's topics that will help also inform the post-workshop consensus paper. Yesterday, we covered two sessions, one examining human milk as a guide for formula development and the other on preclinical models for evaluating novel ingredients in formula. In session one, we investigated the what. What should be an infant formula, at what levels, and in what form? For the first session, we felt it was incredibly important for academia, industry, and government to be on the same level on how we define safety evaluations and educate each other on the processes used to evaluate novel components so we can have reasonable expectations for each other. Regarding levels, we agreed that human milk functionality should guide the concentrations of novel infant formula components, although setting a minimal effective concentration based on preclinical studies could be desirable. The current U.S. regulatory framework does not consider this in safety evaluations. Under the subject of composition, human milk is generally agreed upon as the best model we have for infant nutrition, but bioavailability of nutrients and functional activity of novel components may not be identical to human milk components, and therefore using exactly the same concentrations in human milk may not always make sense. For example, if a component delivered in infant formula is absorbed to a different extent, a higher concentration than in human milk may be needed. Therefore, human milk is a guide that won't give us a direct recipe for how to make infant formula, considering that added components may not be identical. Nonetheless, there seemed to be general consensus that selection of infant formula components should be more based on the function of human milk more so than the specific composition. Regarding form, a majority of audience members agreed that a mechanistic understanding of a functional ingredient is important for selection as a candidate for addition to infant formula, but we don't quite have consensus on how extensive a mechanistic understanding should be or how we define that. However, there was general consensus that structural similarity is less important than overall functionality, and that there are not really exact matches to all components found in nature. There was considerable disagreement in whether it would be advantageous to add a category of ingredients as recommended as a sort of middle space between optional and mandatory ingredients, whether this recommended group could beneficially provide a space for suitable components, or whether it is confusing nomenclature that touches on the subjectivity of risk-benefit analyses, or maybe both, remains undefined. When deciding what components to add to infant formula, there is a strong desire amongst panelists to have an outcomes-based approach, but also considerable fundamental research is limited. We don't have all the answers on human milk component interactions and functionality, including at what levels human milk itself is being consumed by infants. Therefore, though we have established a need to compile data on the many unanswered questions, in practice we have finite resources and can hopefully discuss today where we can draw a path forward for next steps.
Okay, and I'm going to attempt to summarize session 2 with all of its twists and turns. So it seemed like from session 2 that people were in general agreement that the sort of standard rat model, like 90-day toxicity studies and in vitro genotoxicity studies, would be recommended for most, but possibly not all ingredients. We talked a lot about the pig model, and there was some disagreement on this topic, but it did seem like we had some general consensus that a pig model doesn't necessarily need to be sort of a recommended requirement for safety testing, but that it could be useful in particular cases. And part of where we got to with that is that the current frameworks that we have, the decision trees from the past papers like the IOM model, were not sufficiently specific for what we need to help guide studies going forward. So for example, the concept of when a tier 2 evaluation needs to occur based on some triggers that occur from tier 1 studies may not necessarily be fully relevant, because as Claire pointed out, if you find signals that are potentially negative in a tier 1 evaluation, you may not want to pursue any further studies anyway. And therefore, it's possible that we may need to focus on, the pig model may be more important if you have previously identified things in literature that would suggest that, examining some impacts on the developmental systems may be particularly important. But that I think that we really need to create a more concrete pathway for what kinds of functions would trigger different kinds of tests. So I think that that's something that our consortium could do together, is make that sort of defined pathway. If it's an antimicrobial compound, these are the tests that we want to do. If it's an immunomodulatory compound, then this is the kind of test that we're going to do. So I think that's really fruitful areas for us to work on going forward. The other part is that we talked about the NAMs, the non-animal models, or whatever you call that, because there's different ways that people use that abbreviation. But I think in general, we decided that those models can be useful to help us, identify things about maybe mechanisms of action of a particular component, but that they are currently insufficient to replace, standard toxicological models, because they are only representing a portion of a system and not a whole body system. So there's still a lot more that you can learn from a whole body system. And then we also talked about safety and suitability yesterday. And I think it was pretty clear that it's important to keep safety questions separate from questions of suitability, because the safety questions are truly what's being evaluated in terms of GRAS status and infant formula composition from a regulatory perspective, and that questions of suitability may be more in the realm of academic spaces or what companies are choosing to use for their ingredients in terms of like making health claims, marketing claims. And then lastly, we talked about synergy. So do we need to evaluate components in tandem with each other, or do we need to evaluate them separately? I think there was some consensus that evaluating components individually makes more sense from a safety perspective, just because there are so many possible combinations of components that we could look at. But there was also a suggestion that ingredients should or could be evaluated in the context of the other components that may already be present in formula. So you may evaluate it sort of added onto a current formula ingredient composition. In that case, you're including some of these matrix effects that we're thinking about. Okay, so for session 3, we're gonna cover controversies and consensus around clinical endpoints that we think would be appropriate for safety evaluation in infant formulas. And next, we are inviting Vanessa Castagna, who is currently with Saipinion, to take us into our 3rd session of the day. So thank you, everyone.
Okay, I'm going to keep my preamble somewhat short because I think they gave great opening remarks for the day already. But I do want to take a little bit of time. Sorry, Vanessa Castagna, in case you didn't hear that already, from Saipinion. Our company, Saipinion, draws from an international community of scientists who answer scientific questions with certified peer review for both science and for food ingredient safety evaluation. So talk to me if you have any burning scientific questions needing peer review certification. For our 3rd session today, we are evaluating clinical endpoints. We're looking to see if we can expand on our understanding of clinical endpoints for safety investigations for ingredients. This is a nice extension of yesterday's session. So yesterday we heard, as you heard, this great summary from OSU. First, how do we choose ingredients for including an infant formula? Second, how do we do our preclinical investigations? And this is 3rd, when and if we need further outside of growth monitoring studies, further endpoints in a clinical setting, how do we make those determinations for what appropriate clinical endpoints would be, and what kinds of questions can we answer with those clinical studies? I think we can move on to the next slide. So I wanted to start back at the beginning. So yesterday we started our 1st session by looking at these key definitions. Actually, before we even began our sessions, Paul introduced these. And I think it's critical. I mean, yesterday, if you tuned in online or you were here in person, towards the end of that 2nd session, it felt like the waters were a bit murky, a little bit muddy on trying to really parse out what we are talking about with these investigations. So I thought it would be pertinent to, again, review these definitions. Safety and suitability are certainly the core of what we are talking about and trying to differentiate when we are discussing one versus the other. For the purposes of today's discussion, we will be trying to exclusively focus on the top, on a safety investigation, looking at reasonable certainty of no harm for ingredients for inclusion in infant formula. We will, of course, also be talking about the investigation of these ingredients in the context of an infant formula matrix in a growth monitoring study, as that is already pre-established, and we'll see that again in a second. But again, focused on safety, not necessarily on benefits at the bottom. Suitability is a concept that was derived from Codex. We don't really have this as a foundational component of our regulatory system here in the US, although obviously for ingredients to be included in infant formula, there needs to be clear demonstration of their suitability for foundational nutritional elements, but for our purposes here today, for understanding how to improve the functionality of infant formula by including components that, as best as possible, can mirror the function of components in human milk. So we are really focused on these optional ingredients, and although some people in the room may argue with this first term, non-nutrients, we will be focusing on non-nutrients today, though, just to keep our conversation really cleanly in line with what is still unknown, not what is known, which is nutrients or nutritional evaluation. So we are specifically focused on non-nutrients and non-nutrients that have known or putative function inside the biological system, which is the infant. And then the difference between function and benefit, I think, is nice to spend a second on. Again, yesterday we heard these concepts in the very beginning, but then as we entered into the discussions of selection of ingredients and investigation in a preclinical model, it's really hard to keep these distinct, function and benefit. And especially as we begin looking at investigations, preclinical and clinical investigations, if we are talking about benefit, we're looking at some positive outcome. And generally speaking, when we're talking about benefit, if you think about marketing, for example, these would be claims. It's something that you want to say as a positive outcome of a particular ingredient or an ingredient combination. But we want to really focus on function, right? Function is what is kind of the mechanism of action. What is the known or putative effect on that biological system, right? They're the same, but they're different. So we are going to, as best as we can, try to keep our conversation on function, just looking at, we know these things have potential function. They do something biologically outside of standard nutrition. And we don't want to look necessarily at the benefit that this might bring, although we all in the room and hopefully online agree that benefit is essential or really, really important for promoting optimal nutrition in an infant. But we just want to focus on safety for function, safety for functional ingredients here today. Next slide. Oh, I have a clicker here. Thank you. Again, looking at these definitions, we talked about this exhaustively, but just to, again, recenter us here. Essential nutrients, these where there is, if the absence of these would lead to nutritional deficiency. We are not talking about those today. There are very clearly established pathways for looking at preclinical and clinical investigations for essential nutrients. At the bottom, technological additives. Again, we are not discussing those today, although we can bring these up as some examples if they have some potential function inside the infant, but we really are focused on functional ingredients. These are some examples we've talked about in the last couple sessions: DHA, ARA, oligosaccharides, prebiotics, and our favorite in the room is lactoferrin. Thank you. So ingredients that exert functional effect on infant health. Paul talked about this yesterday, and I want to bring it up one more time because it's important for us to, again, frame where is the problem, not what is the established path, right? So the established path is there is an FDA review of ingredients. And again, we're really thinking about ingredients here. So we know already this path exists for looking at toxicology, right? In determining safety or evidence of no harm as best as possible, reasonable certainty of no harm, either through a food additive process or food additive petition process or through the GRAS, generally recognized as safe petition process. Ingredients, once they have been formally reviewed by the FDA, and sometimes, as we heard Barbara mention yesterday, in concert with a clinical investigation, these ingredients must end up in a growth monitoring study within a matrix of infant formula for that formulation to end up approved for market use. This is established already. Standard growth monitoring studies, and we will try to use the GMS term as we talk through clinical trials to differentiate it from non-GMS based clinical investigations. But a standard GMS is anthropometric in general, looking at length, weight, and head circumference to confirm non-inferior growth. And of course, there's post-market surveillance, and some conversations we had yesterday were quite interesting to think about if we can sort of do this right, like get ingredients into an infant formula following the standard path and allow the post-market surveillance to speak to the functional outcome or potentially the benefit of these ingredients once they're in the marketplace and in the bodies of our babies. And last, just my last reminder slide is looking at these two different offices within the FDA agency that already is set up to investigate these parts of the infant formula process. So first and foremost is ingredient investigation. I just put a box in to say what are each of these departments looking at. So the first OFAS is really looking at the ingredient itself. Is the ingredient harmful under its intended conditions of use? And then once that ingredient has been reviewed for safety and is investigated for an infant formula, this is the Office of Nutrition and Food Labeling that evaluates that growth monitoring study. And I just kind of put in very, very brief terms, does the finished formula support normal and comparable physical growth? So those are the established guidelines in place. But today we're talking about the other, kind of what's missing and what's sort of in between here. And again, at the bottom, a reminder that there's really no place currently within our regulatory framework that allows us to investigate the functionality or the safety of functional ingredients outside of standard nutrients or essential nutrients. So today's session is going to focus on this kind of outstanding question: when a functional ingredient actively modulates a developing biological system, so a functional ingredient, are current assessment tools designed to detect the full spectrum of potential consequences? And this is not a standalone question. Obviously, again, we are session 3 of 3, so we are following a discussion on preclinical investigations. And today's first question is, when is preclinical not sufficient? Actually, that was the end of yesterday's session too. When is preclinical not sufficient to completely answer the safety questions of novel ingredients? And when is a clinical trial outside of a standard GMS or as secondary endpoints to a standard GMS necessary? I think I'm going to introduce our panelists next before we get into the meat of this. So we have a few panelists in person, and we're kind of split half and half. The other panelists are online, so we'll have some fun with this one. Hopefully we have fixed some of our formatting issues and we can get good visuals in place. So I'm going to start at the top with John Colombo. John is joining us virtually today. He's a professor of psychology and director of and I'm going to completely botch this, I apologize, the Scheifel-Busch Institute for Lifespan Studies at the University of Kansas. John has conducted research on individual and developmental differences in attention and their consequences for long-term developmental outcomes in typically developing and at-risk populations of infants, toddlers, and children. Susan, can you come up to the stage? So we have Susan from PRISM. She's vice president of consultancy at SPRIM. She's over 25 years of experience as a leader in clinical trials and healthcare, and is going to talk to us quite a bit about e-technology, so looking at decentralized clinical trials. Mona Eskander is also joining us virtually. She is a senior clinical scientist and regulatory expert in infant nutrition with extensive experience designing, conducting, and clinically appraising clinical trials and supporting infant formula. She previously served as senior clinical advisor within Health Canada's Bureau of Nutritional Sciences. Welcome, Mona. Leanne Redman is in the room with us today, I think. Do we have Leanne? Yep, thank you. Leanne holds leadership roles at Pennington Biomedical Research Center and is the academic director of the Charles Perkins Center at the University of Sydney. She's led clinical trials assessing infant feeding, growth, and metabolic development, and focuses on relationships between maternal physiology, breast milk components, and infant metabolic health. Welcome, Leanne. Alan Ryan is joining online as well. Alan is joining us at the top of the next hour, so we do not have him for the first one, but he will be joining us. He's president of Clinical Research Consulting, has over 40 years of experience in clinical research with 175 peer-reviewed articles. He's prepared protocols and FDA submissions as consultant and previously held leadership roles at Sensilo, DSM, and Abbott Laboratories. And finally, but definitely not least, Barbara Schneeman is joining us in person. Welcome, Barbara. Emeritus Professor of Nutrition at UC Davis and previously Director of FDA's Office of Nutritional Labeling and Dietary Supplements. She's received several awards and has been recognized for her work on dietary fiber, GI function, development and use of food-based dietary guidelines, and policy development in food and nutrition. Welcome to all of our panelists. All right. Whoops. Oh, we're already in thread 1. Good. Give me a second. Make sure all the microphones are on. Okay, in session 3, So all of our panelists were asked to participate in a pre-workshop online survey, and they were asked a lot of very detailed questions about clinical trial investigations outside of the standard GMS, looking at expanding on clinical endpoints, et cetera. So as with the first two moderators, we collected the survey results and really looked at where there are consensus, but really more so where there were threads of topics needing further elaboration. So whereas the questions that we're presenting today are not directly derived from the survey, they were trying to capture the general nature of outstanding questions for discussion today and seeing if we can use these to help develop a roadmap. So for this session, as long as we keep on track, we're going to try to get through some of these questions. Start with, is there a gap? What is the question we are trying to answer with today's session? We'll talk about the unit of evaluation, which I think is a really interesting one. Are we talking about evaluating a single ingredient within the context of an infant formula or outside the context of an infant formula in some other fashion? So we'll try to parse that out with that session or that thread too. We'll look at a decision framework. So really that question I asked a second ago, when do we need to do a clinical investigation for safety investigation of novel ingredients? And how do we make that determination? Then we'll get into some detail of looking at specific endpoints, and then how can we do evidence sharing? How portable is the evidence that's generated from these clinical investigations for an ingredient to be used in multiple formulations? So we'll start with our first thread, is there a scientific gap? So the question we're asking here, does a current safety assessment framework capture all potential consequences of adding an ingredient with physiological activity to infant formula? And what we'll try to align on as best as possible are these consensus targets. First, whether a scientific gap exists. Is there a question? Are there unanswered questions that we can explore with clinical investigations? And second, what is that gap? What is the category of concern? And to try to get some definition behind this before we move forward into the rest of these conversations. So to break these down into some specific questions, we'll start with this first one. Is there a category of safety concern that neither preclinical toxicological investigations or growth failure in a standard GMS, that these current assessment tools were not designed to detect for functional ingredients with physiological activity? So do we have Mona?
Yes, yes, I'm here.
Mona, wonderful.
Maybe
Actually,
you can see me, but you can hear me, I hope.
yes, wonderful. So we're going to start. I would love to start with you, Mona, to hear from you if you have experienced in your history with Health Canada instances or occasions under which we have an ingredient with known or putative physiological activity where the framework was not sufficient and you or your colleagues at Health Canada needed to explore exactly this conversation. Have you identified that this is an actual issue within the framework of a regulatory investigation?
Yes, this is a situation has we face this situation. Health Canada has approached this by applying a totality of evidence framework that integrate infant clinical growth study as a primary anchor endpoint, tolerance and gastrointestinal outcome, nutrient status and biochemical marker, mechanistic applicability within developmental physiology, comparative exposure analysis relative to human milk, consideration of potential long-term developmental outcome. For non-nutritive functional ingredient, regulator at Health Canada have emphasized suitability, meaning compatibility with normal growth, metabolic stability, and the immune maturation, rather than relying solely on absence of toxicity. In instances where biological activity is evident, but long-term developmental implications are uncertain, Health Canada has generally taken a precautionary but science-based position requiring infant-specific data. exposure alignment with physiological range, and robust clinical reassurance before permitting use. So yes, there is recognized regulatory category where an ingredient is not toxic in the classical sense, yet the scientific determination require broader developmental and system-based evaluation that has been practically relevant in the assessment of bioactive protein, structured lipid, human milk oligosaccharide, and the probiotic organism in infant form. That said, I totally recognize the huge burden on industry and government resource in case of a more extensive approach is taken.
Thank you, Mona. And I'm going to go all the way to the end of the table with Barbara to see if you can speak from the US FDA side.
Certainly, I'm not going to provide the kind of detail that Mona has provided, but I think when I think back on the discussion yesterday about the standard safety assessment and what's possible with the safety assessment and what Mona has just characterized, to me, the question that comes forward is, does the functional nature of these ingredients raise an additional safety question?
Mm-hmm.
And so if in fact you're designing the safety assessment, how has that been factored into the safety assessment? I mean, I think what we were hearing yesterday is that's possible, but it doesn't necessarily mean it's happened, or it may not have happened in a way that's obvious. Here's the standard safety protocol that we followed. This is how we modified it to make sure we're addressing the safety related to this functional ingredient.
Mm-hmm. So there is a gap in the framework, but so there is inappropriate, no, I would not say that word. There is a lack of clear guidelines for how to investigate these types of ingredients, but there is flexibility within the framework that allows this type of query to be explored. Like we talked yesterday, obviously we're not proposing any policy change. So we're staying firmly within. We understand the existing framework. We understand the existing policy. Is there flexibility within there to answer this question?
Well, I think if you think of it in the context that there's a process, the 90-day notification, as those who work with industry know, you can go to market on day 90. You just don't want an objection letter from FDA on day 91. And so in that sense, yes, the system has to adapt to where we are now in terms of the kinds of ingredients people are looking to add to infant formula. Whether or not there's sufficient flexibility within that 90-day window, that's a harder question to deal with, because now we're just not talking about what has to be done. We're talking about what review system should be in place so that industry knows this is what you have to provide to FDA. So they're just complexities in being absolute.
Let's bring our microphones as close as we can. Just
for okay. Yes, I sat in that back corner yesterday, so thank you for the speaker back there. It does make a difference.
So I know this first question is a bit more centered on understanding our regulatory framework, but for our academics in the room and for industry representation, do any of you have thoughts on this first question? Like, is there a gap? Is there a question that we are trying to answer, and what is the nature of that question? Susan?
Thanks, Vanessa. Let's see if we can get the microphone the appropriate volume. I think there's a couple of things here in that I see the question is specifically about neither tox nor growth. but we spent so much time yesterday talking about growth and how that's the be all end all. And, we're going to know based on growth parameters. I think that if we're looking at ingredients where we don't know as much about them in the long term, this gets to the point of maybe doing a longer study. Once we get past those first 4 critical months, to me as the scientist, it's like, but what happens after, right? We're looking at 4 precious little months in someone's life, but instead of sort of saying that the gap is within the design of those 4 months, I think we're getting all we can out of a 4-month-year-old. But what I'd like to understand is, where are they from an immune perspective? Where are they from a health perspective in general? Where are they from a cognition perspective or neurodevelopmental pathways in 1 year or 2 years or 3 years or 4 years? And I fully understand that that becomes expensive. We do clinical trials and a lot of them, and time is money. We all know that. But I think if you're planning to go into that area and really change how we're, add to structure function relationships for an ultimate benefit of an infant, then you're also held to those standards of that longitudinal type of monitoring. I also think that we know a little bit more in the first 4 months than we used to from a growth monitoring study. I know we'll talk about this later, Vanessa, but one of the things we've been doing a lot of for many years is decentralized clinical trials and within growth monitoring studies. And I think as scientists, we all know that when you have data at points in time, you don't really know what's happening on the days you're not measuring. And I think one of the things that I really like about this innovation is we're in contact with infants essentially the whole time they're in our studies. We're not waiting for the nodes. Are we collecting data on nodes? Of course, absolutely. We're following the GCMS guidelines to a letter. But what we learned is that we have access to a lot of data that has not previously been detected. And my ultimate goal as a scientist is to maximize the signal to noise ratio for both safety and efficacy outcomes. And I think by having these data continuums, if there's something to pick up in the first 4 months, it gives you an opportunity to see it more sensitively. And then I think if we follow longer term, then we're making sure that, by that creative do no harm from a safety perspective. And then also, are we providing benefit? We can be certain.
Thank you. You're introducing new questions. That's okay.
That's what all scientists do.
Yeah, that is,
Yes,
that is what we're doing. Anyone online? John? No, Mona, you wanted to respond to that. Mona?
Yeah, I just need to add that we need to define what we mean by outcome. Are we concerned about transit biomarker changes or durable functional consequence? The core scientific gap may actually be the lack of validated connection between early modulation and meaningful long-term health effect.
I guess does that tie in with this first point that we're talking about? I mean, looking at outcomes versus, the question of, do we have the tools necessary to evaluate the safety of functional ingredients on other physiological systems? Sounds like a similar question, just looking at it.
Well, maybe it's a question more of what is the outcome. In the case of the growth monitoring study, it is about growth. That is the outcome. And I think your points are important that for some of these functional ingredients, growth may not be the outcome that's the most sensitive outcome. And so what is the appropriate health outcome for that particular ingredient?
Yeah, I mean, to me, it seems like if I think about the drug landscape,
efficacy and mechanism of action is done before the phase 3 clinical trial to evaluate safety. So I would hope as a clinical investigator that the mechanism of action and those sort of studies have been done before, because once we start thinking about things to do with neurological function and even immune health and the gut microbiome, then we start talking about a narrower set of clinical centers who can participate in those sorts of trials, because Mona mentioned about balancing the totality of safety and outcomes with burden. And that's burden for obviously the infant, but the caregivers as well as the clinical sites, and keeping people in studies for the long term, which is also what we're hearing in this session. Like 2 years, 4 years becomes extremely difficult once you start, layering on the top of a growth study much more comprehensive measurements. I have an example. I don't know, several years ago, 5 years ago, I was doing a study for Nestle, and this is Nestle from Switzerland, and they had a new formula that provided over and above recommended levels of 5 different nutrients. It was DHA, ARA, folate, a whey protein that was fortified with other components, and they were really interested in myelination in the brain. We were doing MRI scans, brain scans in infants, and the duration of the trial was from birth to 1 year. Now, you can do pretty well with NAP scans in babies at 3 months of age, but try to do a NAP scan or an evening scan on a child that's 1 year old. And that's where the outcomes are being properly evaluated, is at 1 year of age. And so, to have a final sample size of 68 babies, we had to enroll 3 to 1. So, it's 170 infants. And at the end of the day, there were 2 sites in the US that could participate. So I think we need to be really careful what we're sort of asking for and what we're thinking about here. And again, I'll go back to where I started, which was sort of the drug landscape. And I'm wondering whether there's an opportunity here to do this efficacy work, establishing mechanism in the human infant prior to the growth study. And it can be part of the package as well, but it's a smaller number of participants, and it's a very, very focused study on one particular outcome.
So we will be talking about your experience in a little bit later on when we're talking about feasibility of incorporating different clinical endpoints in our trials. But feasibility is a very solid point that we'll hopefully keep as a context and a framework around our conversation today. Yes, like doing a lifetime study, actually, which is what anecdotally I've heard some people talk about, would be ideal, but there's no possible way. It is not feasible. It's not practical. So now, what within the framework of practicality should we be proposing?
But I think we also need to build on the experience you talked about. That I know Paul's going to have a heart attack when I mention the word claims, but we do know that part of adding these then will lead to the use of a structure function claim. And the policy around structure function claims, FDA has issued a draft guidance on structure function claims. But the reality is the manufacturer has to have the substantiation for that claim. And that's what I think you're talking about, is the substantiation that they have. That's not provided to FDA unless there's an enforcement action. But what I'm hearing is the data are there, the evidence is there. If challenged, most companies would be able to produce the data. So I'm not saying it should be required as part of the notification, but how does the fact of its existence help in resolving this dilemma? That's probably the best way to position it.
So I'm glad you're pulling claims in, and because I'm thinking as we're talking, again, going back to the central question, what is the outstanding issue? So let's put claims aside and go back to the science, and we can open up to this room too. I guess this is in part following the end of session 2 yesterday, preclinical investigation. Again, what questions are insufficiently answered in a preclinical setting on safety of functional ingredients where we see a need for additional endpoints in a GMS or a separate clinical trial? specifically to investigate those outstanding questions of safety of functional ingredients. So, I mean, we've talked about different physiological systems like the GI tract, neurocognition, right? Metabolic, potentially metabolic activity. Like of those, you know, we talked a lot about the piglet model yesterday as being potentially sufficient for some of that investigation. And we hear from Mona from Health Canada that clinical investigations seem to be pretty foundational for gathering complementary information about these different physiological systems. I guess for the US, do we feel again that these, like, do we agree that neurocognition needs to be investigated in some type of clinical trial, whether that's a secondary endpoint in GMS or a separate trial altogether for an individual ingredient to confirm or affirm the safety of that ingredient because it has potential impacts on cognition? Or do we feel like that preclinical package, again, just for the ingredient alone, should be sufficient for its safety determination? John, I would love to hear from you too, because of your experience working with these types of ingredients.
Right. This is, can you all hear me? Yeah.
Yes.
This is a different, you know, you're walking the line between function and, as you've defined it, function and benefit. A lot of the neurocognitive effects of early nutrition have been considered within the realm of benefit. But is the lack of the benefit now a safety concern, is how I see this. And so, you know, and in the preclinical level, that adds a whole nother set of complications, because there are very few preclinical outcome measures that have been used that are actually directly applicable to human, especially early in life. This is the question itself is fraught with a whole bunch of definitional problems, as I see it. You know, we've used historically, you know, adverse events and growth as an indicator of safety. That seems to be a pretty low bar for me, but that's, for what it's worth, that's what I think.
Yeah, I guess anyone who has that experience, John and others in the neurocognitive space, like what would be the safety concerns that you are looking for in a clinical trial that would not be evident in a preclinical model?
Well, developmental delay would be the clear indicator. Developmental delay as measured by the standard global. Someone yesterday said, you know, we have great tools. I would disagree. And so that's another problem that we have. But if we find, for example, a developmental delay on a Bailey or on a Mullen or something like that, that would be a clear indicator of a safety problem.
I guess to challenge that a little bit with the discussions yesterday, and we'll use our good guinea pig Paul, who stands here and says we should be able to detect that in a toxicology study. We should be able to see the trigger, that it will have cognitive effects. So when would there be a case, anyone in the room, where you would not see a trigger in that toxicology study that you may see when it gets into an infant? Again, trying to say, like, where is the need for a clinical trial? When would that trigger be necessary? Do we have any thoughts on that? Bert?
I would refer to the very impressive work that John Colombo has published, where he showed that changing the ratio of DHA to arachidonic acid changed the outcome at age 5 years. And he basically showed with an inappropriate DHA to arachidonic acid ratio, the developmental outcome was attenuated, which I think is a really fantastic study, John, that you did over many years. And there was no such signal before in any preclinical study.
You get an A. Good job.
So I would then ask the question, how would that fit into a determination of whether or not you can go to market with the preliminary information versus waiting the 5 years. I mean, that really is the dilemma of, is there enough that you can do it when you first come to the realization that this is important, or did you have to wait 5 years?
Yes, indeed, that's an unsolvable dilemma. And I guess in the real world, one has to find a compromise somehow. But we've all learned about the term of the 1,000 days, and we've seen many examples that things that happen early in life can have a lasting impact many years later. But we just heard doing such studies is difficult. We just completed a follow-up investigation at age 20 in children that we randomized on different feeds in infancy. And that's I can tell you, a really challenging thing to do. It's difficult to get the funding, and it's difficult to follow children prospectively for 20 years. We would all love to have such studies, but you can't expect that somebody who has an innovative ingredient waits 20 years or 25 until you use it. So it's, in my view, it's an insolvable dilemma, but it obviously depends on the ingredient you're thinking of. So let's think of a worst case scenario. If somebody would come and say, I want to add IGF-1 to my product, we would all get concerned because we know IGF-1 levels have been associated with cancer risk, breast cancer, other cancer risk. So that's a situation where probably any regulator would have a red flag and say, we'll need to be very careful here. With other ingredients, when you have less level of concern, you basically have to take a decision.
Just had a question, Kristen from DSM, with that DHA ARA example that you gave. So what happened is if it wasn't the correct ratio at 5 years, the babies didn't have the benefit, but it wasn't like they were developmentally delayed, so does that mean that it was unsafe, or does it just mean that we no longer saw the benefit?
That's a great question.
Yeah. So John?
John?
Yeah, I mean that's exactly right. That's what that's the point I made is that we did a dose-related study on DHA, and at the highest dosages we saw reduction of benefit. We did not see developmental delay anywhere, but we tried to explain what it was, and working backward, we basically were able to show that was more of a physiological study in that as we increased the amount of DHA that was given, we saw a decline or a quadratic decline in ARA in the system, which suggested that there was some interference with the metabolic pathway. And now I'm talking way past because I'm a psychologist, but that's is that a safety issue, or is that a lack of benefit issue, which is what I raised at first.
Mm-hmm.
I think that probably starts to get into the benefit space. I mean, you didn't see a difference in that group that had higher DHA relative to ARA versus the control, which is the standard that was on the market prior to the introduction of DHA. So that study was really about trying to optimize the benefit and like a further exploration of kind of optimization down the line. And, that trial was long and expensive to run, and it wouldn't have happened without that, all of the foundational studies that took place before that one could be run, kind of getting the support to run that trial wouldn't have happened without the initial work and even having the product on the market probably.
Okay, this is a very interesting discussion, not where I expected it to go.
I wanted to just follow on some of this discussion because, Christine, I think the issue you raised, to me, it begs the question of what is the risk of adding it? What is the risk of not adding it? As opposed to benefit harm kind of thing. And when you look back at that data, could you make an argument that the risk of not adding it was greater than the risk of adding it? I mean, Bert, you're nodding your head. I don't know if you agree with that or not, but it seems like the questions become different.
And Barbara, just to respond a little bit, and also the discussion you were pointing out is that, you know, there's reasonable certainty and there's absolute certainty. And we know absolute certainty is essentially impossible. And for the most part, we function on reasonable certainty. So I think that, to your point, you know, I know we all joke about we could have the "it depends" t-shirts, but it's true. You know, it's funny because it's true. You know, if we're talking about ingredients where we have a reasonable certainty that it's safe and they're well accepted for a variety of reasons, that's one thing. But I think when you start talking about what Leanne was saying, where now you've put 5 novel ingredients, we don't know how these things interact, you're doing MRIs, that doesn't feel like we know what we're talking about yet. It feels very uncertain to me. And I think there is a balance where, you know, you kind of, maybe you do some cognitive assessments. You know, we talked to John, and we're doing those cognitive assessments and we're watching and things are going okay. And then you're able to kind of ease up on allowing these things to come into the population at large without, because to your point, Barbara, if we all wait 5 years, nothing will happen. But I also think, for me, I constantly ask myself, where is the line between some of these things that we're talking about and a drug? You know, and it comes, all right, when you add 5 new things that we don't know how to, when did that, what defines, I know the definition of a drug very well, but when we're talking about modifying brain function, how, like, this doesn't seem like food to me.
Although the argument, I'm sure, would be, no, it fits into the structure and function. You're helping to maintain the structure and function of the body. Yeah, it's how you characterize the intended use of the product. Is it brain function that's over and beyond, that's correcting a problem, or is it maintaining the structure and function of the body?
Like comparable to what? That's kind of what we're talking about too. Comparable to infants on infant formula, or comparable to human milk fed infants. Right. And I'm guessing we're talking about the latter.
Yep.
Right. Oh. Mm-hmm. And in which case you could potentially argue everything we're talking about, that where we're discussing putative benefits, it would not necessarily be benefits in and of itself as it pertains to like a drug, but more so attaining outcomes closer to those of human milk-fed infants as the bar.
Right.
So Vanessa, in that intro slide, you had like definitions, right? Like there was a definition for suitable, safe, beneficial. And then under the definition of safe is a reasonable certainty of no harm. So should there be a definition of harm?
That's exactly what we're talking about here. That's exactly right.
Paul has a definition.
I don't have a definition, but I think I'm trying to synthesize everything that we're talking about, and I think a definition is important. And I think one of the things we're missing here is, when you're evaluating the data, how you evaluate the data will get you to the conclusion. So if we're talking about, here's what formula without the ingredient has, and that's what the function is, and then you're talking about human milk, getting closer to human milk from formula without the ingredient would be, you could call it a benefit, but you know, getting closer to human milk, that's better. Getting further away from what human milk performance would be, that would be getting worse. Is that adverse? I mean, you could argue that, but I think the DHA example is a good one where, okay, maybe with DHA and ARA at the right level, you're as close as you're going to get to human milk. But even if the ratios are a little bit off, you're still getting closer. You're just not getting all the way closer. So is it adverse? I think you could argue it's not adverse. It's not as good, but it's not adverse. So what is harm? Like, I will throw it out there, like harm is getting further away from function of human milk.
Yeah, and if that's the case, going back to the same question, when do we ever envision a scenario that you would not detect that in a preclinical study, where you get into a clinical trial and suddenly you are harming infants? Is that the genuine concern that we were trying to address?
Which is the safety.
Safety.
Yeah, the safety side of it. Yeah, from the summaries that were going on, I had written in my notes, what is the justification for adding a novel ingredient? Sort of, we say, okay, it's in human milk. That might start you on the path, but it's not sufficient, not enough. So it is there. And then you might ask, well, what's its function in human milk? And then if you know what that function is, what's its ability to mimic what happens with human milk? And then I find the way you characterize it, if we say infant formula is here, human milk is here, we're not going below where we currently are with infant formula, and we may go higher, closer to human milk. I don't know if you can do that in a way that you can see it in experimental data, but that paradigm sort of helps you think about why would you want to proceed with getting these things added.
Well, I have a lot of further questions, but I still feel we have not fully identified what it is that we are trying to answer with all of these follow-up questions. Do we? Oh, Mona. Oh, I didn't see. Thank you, Mona.
Yeah, I just want to say, going back to the story of the DHA and the ARA, it is supposed to be beneficial because they are present in human milk in a certain ratio. And we know that DHA and the ARA are accumulating in the third trimester of pregnancy. So we know it is supposed to be doing something. The risk of not adding them, we are depriving infant from this. And this also takes us to the story of preterm infant. We know that preterm pregnancy miss the last few weeks. That's why for preterm, we always allowed to add more DHA and ARA to preterm infant formula. So we understand that it is doing something for the infant. If we are not adding it, we are depriving the formula fed infant. So basically, we should think of adding ingredient either to mimic the composition of human milk or the function of human milk.
Yeah, I think that was a lot of what we were discussing yesterday. And the answer is yes to both. Maybe potentially prioritizing function over the specific composition due to difficulty in replicability of the components of human milk, and also our lack of understanding of the full nature of the composition of human milk. But certainly functional outcomes, and I think we mostly have aligned on that in this room and our online community, that looking at, I mean, that's why we're here, looking at the functional outcomes and the disparity between the outcomes of infants exclusively fed infant formula versus those on human milk. That gap still exists. And so that's what we're trying to address with this conversation of how to close that gap and how to select the appropriate ingredients for that.
But I think the issue that Mona has put on the table is, are you doing harm by not including it? It's sort of that question of which way does the risk fall? Is there greater risk of including it or greater risk of not including it? And I mean, I'm not an expert to say whether that's a correct way to look at it, but I think that's what she's putting on the table. Mona, did I interpret that correctly?
Yes, yes. Yes,
And-
that's why, because we know it is doing something. As I mentioned, I know we are not discussing preterm, but we are even allowing the addition of higher level in preterm because we know preterm miss the correct accumulation, the level of accumulation during pregnancy.
And my guess is most infant formulas on the market include DHA and ARA at this point.
Most of them, but it is not yet mandatory in Canada. But I guess most of them, yes. But
Jody Butler.
we have actually a paper on the level recommended for addition to infant formula, and we requested that the ratio should be either 1:1 or 2:1. I mean DHA high. And in Canada, if we allow the addition of DHA, ARA should be also added. Otherwise, they will compete metabolically.
Mm-hmm. Thank you, Mona.
Jody Butler from Nestle Nutrition. To me, this is not about safety, this example with DHA. It's about having enough evidence to where now you say it must be in there, which is a different question, and I think that has happened other places, but that's the benefit of doing all these studies is then you find out eventually that we have enough reasons to believe that it shouldn't be optional anymore. It should be mandatory to add DHA. I still don't hear a firm answer to this gap. What I hear, I haven't heard an example of where we didn't identify a safety issue from a preclinical model. So just from listening to it, I haven't heard an example. I feel it's more of a, not a fear, but a question mark is as we add these functional ingredients, we're not confident that perhaps we're not confident enough that there may be something happening biologically that we're not capturing in an animal model, but I haven't heard an example where we have identified a gap clinically that wasn't flagged preclinically. Am I wrong?
I agree with you.
It's more of just a concern, underlying concern, but not a real example of where that's happened.
Right. Oh, yeah, just kind of wanted to pick up on that point, Ashley, from you, Johnson. What is the purpose of the data being generated? You know, there's an initial piece of putting an ingredient on the market where you're evaluating safety. You're not making that like risk benefit assessment. But later in the process, you know, there is also a regulatory component of setting, you know, infant formula standards of composition. And I think, you know, that piece kind of picking up on the evidence you were pointing to, Dr. Kletzko and Colombo, that trial helped to influence setting expert recommendations for the composition of formula once we had kind of amassed a totality of evidence and a collective understanding that this component was important, and then, you know, might go on to influence regulations on the standard composition of infant formula, but was not used up front for that initial safety determination to put on the market. So, I mean, similar to what you were saying, but just thinking through which data apply to which part of the process and the timing of that process.
Dave?
I had two thoughts, like just playing like the devil's advocate here.
Of course.
Of course. So first is like the topic that I brought up yesterday about like the concern that was previously mentioned about autoimmunity potentially. Like in that case, I could see, I know that yesterday we talked about like whether or not that's a valid concern or not, but it did kind of seem like that could be a sort of a case, which is what we're looking for here, in which something beyond the standard preclinical safety testing may make, you know, it may make it relevant to do a human study. The other one that I was thinking about was Bert was bringing up like IGF, for example. So like things that might have a hormonal effect that could affect the development of, you know, let's say like intestinal differentiation, or, you know, like some sort of organ development. And you could say that those would be identified via a rat study, particularly if you use like an infant rat model as opposed to an adult model. That's possible. It's also possible that the model may not be sufficiently similar to a human to be able to detect the effect. Like, for example, let's say that the digestive capacity of the model is not the same as the human, right? So let's say that hormonal component, let's say it's like a peptide component, let's say it's more digested in a rat model than in a human model, and therefore the potential effect is not as high for that hormone to exert an effect on the tissue. And so in that case, it is possible that you could miss some sort of effect. That's a devil's advocate.
Yeah, so to use Barbara/Codex's brackets, I would put brackets around recombinant human proteins. I'm sorry, Helena. Sorry. Just because this is a novel technology that's allowing us for the first time ever to produce ingredients that are identical in nature to human milk components, but from a different production methodology. We're not isolating them out. We also are unethically prohibited from isolating components from human milk for testing in infants. So there has typically or historically been lack of precedent in us investigating these ingredients in the context of infants. So I think from my perspective, my limited perspective, I think that is somewhat safe to put brackets around recombinant human proteins or other recombinant human components for infant formula and investigating their safety. And potentially that would be one, breakout workshop where you investigate the benefit of clinical trials for those types of ingredients. And the second category, I know you mentioned hormones specifically, but maybe what we're talking about is like receptor specificity. So if we have a component that we're isolating from a different species or that we're generating through a recombinant source that has, species specific interactions. Recombinant human obviously is one major category. We know that binding affinity is much higher for human proteins as it is for any other. Bovine, same, like most bovine proteins or components have very similar interactions with human receptors, but not the same. So there's less efficiency. And as we go to different preclinical models, the piglet model, the rat model, again, specificity does matter. So potentially, if we have that as, again, like a bracket, we can come back to saying receptor specificity matters. And if there is very clear differences that you would see in an animal versus what you would see in a human, that might be a category of ingredients that you might need a clinical investigation for due to the lack of clear evidence in a preclinical model.
Hey, I was just going to echo, this is Ross from Helena, that, to your point, Vanessa, yeah, and agree. And from 2008, there was a panel that looked at human recombinant proteins. They agreed a clinical study would likely be needed to assess some of these safety questions that were brought up. And then another panel that Helena put together, to kind of cross-validate that recommendation. A group of immunotoxicologists, they also agreed that for human proteins to assess this question of, immunogenicity, that a clinical study would be needed. The one question to consider is if you run a clinical study in an adult population where tolerance in the immune system, and I know enough to be dangerous here, but just kind of broadly the question, if you don't break tolerance in an adult who, whereas in the infant where they're in a more tolerogenic environment in their gut as their immune system's developing, would that be sufficient to not have to do that in an infant, given that if you show, you didn't break tolerance in an adult? I think that's maybe a question, but I think broadly agree with that recommendation.
Now I would love to pose that question back to our experts in clinical trial design. Is
But-
there a
but I
place for
think-
human adult?
My understanding of the question is it basically comes back to a safety question.
Right.
Can you use exposure in an adult to extend that safety assessment to infants. So
Yeah.
we're on the clinical side up here. And I keep looking at these questions, and I read those questions and I say, yesterday's panel said, yes, we have ways to do this. Now, maybe we're not making it obvious that that includes these functional components beyond just a, traditional safety assessment, but maybe that's what needs to be clear, is how is safety of these functional ingredients being addressed in the current assessment tools?
And
I mean, Paul, you're nodding your head, so.
Just to add on to what you were saying, what we resoundingly hear is sponsors are looking for this guidance. They want the roadmap to follow. It doesn't exist. It's not well defined. And I hear us struggling with definitions ourselves. But I think a wonderful start to addressing, let's call it a scientific gap, is that when we are entering this category of possible structure, functional claims or relationships, we have the expertise within this room to put the rules in place. We can talk to John and others about, if you're going down a neurodevelopmental pathway, what cognitive assessments do we recommend be tested, and at what ages, and what are we talking about, and for how long, and what falls into, if we're talking about gut health, how do we measure microbiomes, and what are we chasing, and what are the standards? And I think it's a place to start where we can start to say, these are the things we expect you to test at a minimum if you're going into these systems. And then the field can start to align with, there's a paper out there that says we should look at this for brain, and this for gut, and this for immune. And the standards will begin, we can always modify, but we don't have even kind of those basic blocks. And everybody's a little bit doing their own thing, at least is what I'm saying.
So you're pushing us to keep this conversation going and finish talking about these endpoints. We're kind of stuck in this very early kind of water churning stage of, is there a question that we're trying to answer? And I think we're getting a little further along. Sharon.
Well, I'm going to change the topic, but I want to respond to the IGF-1 question, because way back in 1997, which in PubMed is ancient history at this point, but we were very interested in IGF-1. Actually, Doug's done a lot of work on it. So we actually took newborn piglets within 2 hours, fitted them with umbilical, portal, and arterial catheters. So again, and gave them iodinated IGF-1 orally. And we said, how much could we detect in the blood over 4 hours? 0.205% of the circulating pool. And so this concept, so again, I think these are questions, we've done this for comparing intact soy protein with soy hydrolysates. We're doing this now with exosomes to see actually how many, which exosomes in milk their microRNA package is being absorbed. So if you do have these questions, I think this is where you could do targeted preclinical studies to answer some of the questions, Dave, you're asking, if it's something that wasn't there in the baseline. Because, I don't think a lot of the hormones in milk, and plus a lot of the activity of IGF-1 on cancer is autocrine and paracrine, not endocrine, because 99% of the IGF-1 in serum is bound to binding proteins, which inhibits its receptor activity. So, again, I think it's understanding the mechanisms and the basic research, but, there's a lot still we can do in the preclinical space to get to these questions.
And again, like drugs, the mechanism of action, proof of concept, is at the preclinical stage, not in the human.
Yeah. Dave?
Follow up for Sharon's point. One thing that brought up for me is that, like, for example, when you're talking about absorption, one thing that could be different for a pig and a human could be absorption, right? They have, pigs have a less quick closure of their gut, right? Because they need to absorb IgG from human milk or from their own parents' milk, right? Not from human milk, from mother piglet's milk, I guess. Whereas humans don't do that. We have like a different mechanism, right? So it's possible that we have different levels of absorption of compounds.
Well, gut closure is about 36 hours in the piglets. So if you wanted to avoid that, but we actually did it at this time point because we're saying we want to see what could be the maximal absorption. So yeah, it is different because there's no prenatal transfer of immunoglobulins in the pig or the cow or the goat.
So to follow up on your point, Dave, if you do have a protein or a component with where ADME is different in a human versus any of our models, piglet or rodent, et cetera, would you propose that would necessitate, require an infant trial for that ingredient? So if ADME is different, if it's species specific, you're the one that asked. I have to ask it back to you.
I don't know the answer. Sorry.
Paul?
I don't think every time. So I think you'd want to look at, yeah, it depends. So, I guess from Team Rat, I'll make the proposal that, the question up here of do we have a gap? I think I will take the strong toxicology position of over the last 50 years, we have developed extremely robust preclinical models to evaluate, to get to reasonable certainty of safety for ingredients in food, pharma, agrochemical, et cetera, that establish safety of these substances without a clinical trial. So do I think that there's a gap, that there are some endpoints where only a clinical would be appropriate to assess? Maybe I'll take Ross's example out as maybe one, if we determine that's even a relevant endpoint, honestly. But I think the answer is no. Like, I think you should be able to get through these preclinical studies. And I'm also trying to think, if we're talking about going from function of infant formula, are you getting closer to human milk? Like, I don't care above the line from formula without the ingredient up towards human milk. Like, that's all benefit. That's not what I'm talking about for toxicology. For toxicology, I'm talking about going below the line. And going below the line, like, that is going to have some adverse effect on mammalian physiological systems. The models that we're talking about, OECD 408, all these rat studies, the piglet study, I mean, those should be, if you're having a physiological effect in mammals, like, that is adverse, you should be picking it up in these models. They are very robust.
Well, and I don't know how many compounds are studied and then rejected because they don't make it through that. And that may be something we need to understand, that these systems kick out what's not going to go forward. Is it the majority of compounds that are studied? I don't know.
Yeah, and that goes into what Claire said yesterday. So if we were studying a food ingredient and you had liver toxicity in a rat study, like, okay, we're moving on. Like, and how many of those get published and how many do people ever find out about? None, because it goes into the trash heap.
I can, since Claire's not here, I'm her surrogate. Hi, Jen Simmons from Spherix Consulting, filling in for Claire. I know I've seen this where when we have a new ingredient, whether it's for infant formula or not, we usually start with the OECD protocols for genotox. And those are done some are done in bacteria, like an Ames assay. Others are done in vitro with cells in a dish. There are some that can be done in mice. And it's only if you have complete negative signal from all of those at doses that are feasible that you would go on to even do like a dose finder in a rat before you do the 90-day. So the dose finder in a rat's a 14-day. And then sometimes, depending on the thing, you do an acute dose study before you do the 90-day. So before you even get to the rat study, you already should have a fairly good idea of what you're looking at. And then if you get to the rat study, which is exhaustive, because I've reviewed many of these studies, and these study reports are very long, and they're looking at everything in these rats. It's only after you have a negative response from all of those that then you get into, should we do a piglet study? And for some of these, the answer, I think, even though I'm on Team Rat, piglet studies are very useful and you can address a lot of these questions. And then I have in my notes here, like, we're trying to a reasonable no harm. So then where do we go from reasonable towards the lofty goal of absolute, as you guys already said, is unattainable. So I want to stand up for the toxicologists that I am not, but that I work for, and say that, yeah, these are very robust studies, and there's no way that we would get that far if there was a chance that there would be below the Paul Hanlon threshold of regular infant formula.
That's on record, Paul.
Sorry, Paul, we're giving all the credit to you for all the things.
But then my question would be, when you're adding one of these functional ingredients, and you want it to be suitable for infant formula, how do you modify that toxicity testing? Is it the same thing, or is there something you need to pick up specifically to make sure that you're not carrying forward something that could cause harm?
This makes me think something about Leanne said earlier about how in drug you have the mechanism first. And yeah, I come from a background of like cell biology and cell signaling, and you never go forward without a mechanism. And so I think that if we have an idea, a hypothesis of what the mechanism would be for these functional ingredients, that then we can add an arm to a 90-day, or do the next generation toxicology study that Paul was saying, or design an arm for a piglet study before we get into the humans to address some of these things. We can be thoughtful when we're putting together the safety package. And if there is still a concern, like maybe there's differences between animals and humans, then maybe, yeah, adding an arm to the growth study in the infants when you put it in the infant formula. I think that's with the tools that we have and the framework that we have, that's the best option. That's the way that we approach things at Spherix Consulting, Mrs. Jen.
So maybe I'll add one more example. So when I went through and read all the GRAS notifications for infant formula ingredients, one set of substances that there's a lot of notifications for are probiotics. So lots of organisms. So probiotics are a very strange set of substances. You don't run gene tox studies on them because it doesn't make any sense. But one study that a lot of them did do was, and I don't understand it because I'm not into these studies, but they had intestinal cells and they'd incubate them to see if there's like changing in the binding of bacteria to these intestinal cells. So it's backing up when Jen says, like, yeah, like if you know there's something about the ingredient that would say, actually, we really want to pay attention to this focal area of where it may have an effect, there are models that you can use preclinically to check that out. Maybe I'll turn over.
So I can, from Menke from Novonesis. So I am from a probiotics manufacturing company, and I think you're referring to the intestinal cell adhesion assays. And that's to show that the probiotics can actually attach to the gut. So it's more of an efficacy type of study as opposed to safety. But back to the question on the tox studies, and do you need to modify it for functional ingredients? I mean, there are challenges to doing rodent 90-day studies, like for macronutrients, for instance. You can't dose high enough lipids. Yes, so there are certain classes, but those things are not considered inherently toxic per se because they are macronutrients. But to also the question as to, and I think this was raised yesterday too, like in pesticide world, for instance, it's very common to see adverse events in a preclinical tox package. But we're talking about food ingredients here that are modeled after something that is in a food that people ingest safely. So it almost sets like the bar, the starting point is it should be safe.
I would just like to add on to, before we get off of the topic of some of the comments made about rat and pig models, even though preclinical was yesterday. And I'm very much looking forward to clinical day, but I can't help myself. I'm going to dive back in. I mean, I'm coming from a perspective of having worked with mice and rats to the tunes of thousands and thousands over many, many years, and I understand what you're saying. I also understand the conversation we were having about the piglets yesterday and similarities to humans. And I had just a couple comments. One is, I don't think we're as sure as, just do everything in the rats and that's fine because that's what we've always did. There's advantages to rats. It's so inexpensive. It's so well controlled. We all know exactly what we're doing. All the rules are written and you run the recipe, right? It's like getting a pre-made cookie and just shoving it in the oven. It's that easy. Like we know exactly what we're going to get. But a lot of what I do is ask, like, where fundamentally have we learned more or grown from our origins? And like, I have to ask the question, if the answer to the question yesterday was, we've already made the rule book for piglet studies. We already know everything there is to know and how we would do that. And it doesn't cost anymore and they're readily available. How different would this discussion be? The discussion is because the science with enteroids and piglets and so on and understanding the difference between neonates, it's further along than we are practically. And I think therein lies our struggle because we all would say, well, listen, if something is 90% similar and the rodents are not, the 90% thing would be the better standard. But we're nowhere near being able to apply that standard, so we struggle. I come from an oncology and cardiovascular background, and we used to say, we'd walk around cheering all the time because cancer was cured in rodents over 20 years ago. There are no rodents left with cancer. And there's fundamental differences where these models break down. And I think the struggle is how do we balance using the rats, which give us this great, fast, consistent 500-page report that we can be sure of, with the translation of that to a human.
Bernstein, non-research. So if I still look at the question 1 and 2, we have 2 topics addressed today. I think one is we come with a scientific hypothesis. We have also the scientific basis, which is also considered for the safety evaluation. So we come with a concept mimicking human milk with a functional ingredient. If we have concerns like a hormone function, we would take extra caution because we expect some long-term effects. Is everything covered by tox data or growth data? Perhaps not. Because we had this example of arachidonic acid and DHA, which is not covered by classical growth data. It's only on longer term. So that means long-term effects are not fully addressed, and perhaps they should. The second one is the biological activity, and that's not just about safety, it's also by an extra effect. So we heard this example of myelination. So of course you cannot detect a myelination in a classical growth study where you just look at head circumferences. So the myelination assay you gave, I think, is really intended to show there's a potential extra brain benefit. That is per se a positive test. So it's not about safety issue, it's just to show an extra benefit. And I would expect in those extra biological tests, which are not just growth studies, of course, we would observe if there's a negative effect. And then it, of course, adds to the adaptation which is needed to use an ingredient or not. But the intention is there's an extra functional ingredient which brings benefits and not safety concerns. We do these extra studies, and if those extra studies show negative effects, we have to react. But I think it's the intention of the industry and the suppliers and also of the academic parties of the hospitals involved to intend with all the scientific data that this is per se a beneficial ingredient. In the worst case, it should be not active, but not of a safety concern. So I think we are covering this question 1 and 2 different aspects. One is the pure safety, and the question 2 is an add-on biological effect we are all intending, because we talk in this session about extra functional ingredients, and they should come always with a benefit without any safety concern.
Let's get into our first Slido question. So this is an opportunity to hear from, of course, those of you in the room, but certainly our virtual audience participants, to really answer the same question we're looking at already. Do we think the current infant formula safety assessment frameworks are designed to detect safety signals from non-nutritive ingredients with physiological activity? Wow, so far, quite a response. Okay.
Can I make a comment while we're
please
gathering the numbers? So I appreciate your comment, and it seems it brings me back to the issue of claims, because the companies who are intending to add this, and I don't know where in the process the research gets done to see if there's a benefit associated with this ingredient, and then the safety of the ingredient. I don't know how all those timelines converge on each other, but at the end of the day, there is data linked to the benefit that may be relevant in trying to understand the safety.
And I think part of this is also, if you think about a study where you're looking at function of an ingredient in a clinical model, so is it a safety study or is it an efficacy study? The answer is both. I mean, you're going from standard infant formula, the polyethylene line, like, are you going up and getting closer to human milk? Okay, then it's a benefit study. If are you going away from human milk? Okay, then it's an adverse effect and it's a safety study. And when we have been describing these studies, that's kind of where we have gotten lost a little bit is, oh, don't tell me about benefit, but is bringing it closer to human milk function, isn't that also, even if it is a benefit, isn't it also a demonstration of a lack of a negative effect?
We're just talking about totality of evidence then. I mean, it doesn't hurt to demonstrate that it's beneficial in an infant or brings outcomes closer to that of human milk, but is it required for demonstration of safety? Yeah, Dan.
I think similarly, though, I come back to the question of what is harm. And we talk about benefit when we think about the other side of that question is a lack of benefit. And so that list of things that are detrimental compared to breastfed or human milk fed infants that we're not adding, I think we need to talk about traditional safety and harm, but then also sort of what is the harm of not adding something that we know has a benefit.
Lots of sighs.
I know. It's the risk analysis.
Cypress, can I advance this? Oh, there we go. So we were supposed to have aligned on what is the question we are trying to answer. I think maybe we have gotten to a couple of brackets of if Helene is now out of the room, if Helene, I'm just kidding, if human ingredients from recombinant host are the query, the components we are investigating, then maybe we have a place here. Or if species specificity is important for that evaluation, for receptor interactions, et cetera, maybe there is a place for further clinical investigations for safety evaluation. So far, that's all I've heard as the real maybes that are still the gap. Do you have more?
I can create another maybe.
Okay. Yeah. Let's hear another maybe.
I was just okay. This is very hypothetical, but we were talking about all the components in human milk may not necessarily be beneficial for the infant, right? So, for example, human milk can have the presence of different bacteria after expression, and some of those are not always perceived as beneficial, right? Or, for example, like you could find a bacteria associated with human milk and think, oh, we should use this as a probiotic, and you could test that for safety in a rat study, but perhaps something about the structure of the human intestine, the proteins and glycosylations that are present in the human may encourage or discourage growth in different ways of that bacteria, which could affect its potential impact on safety, right? So just I don't know. I'm getting hypothetical here, but you know, that's another possible area. I don't know about like past examples of studies with probiotics, whether prior to getting acceptance they use clinical studies for part of that safety evaluation. I don't know. So, yeah.
So when I first saw this consensus target, my thinking is, I think we've had a lot of discussion about the issues. We could do a nice laundry list of all the issues that have been identified. And I think the one area that we perhaps come closest to a principle is that notion of, you know, are we moving toward human milk or are we moving away? And the goal is to move toward human milk in terms of improving infant formula. And so that I think we all agree that that's a reasonable place to go. And so then the question becomes, how do you do it? What do you invest in? Because you have to address those issues to live by that principle. So I don't have an answer, but it's a way of just sort of what have we achieved so far.
Mickey, did you want to respond to the microbiome question?
Sure, it was a big hypothetical, but I think if I follow what you're saying, and just for historical precedence, a lot of the probiotic strains currently used in infant formula are like lactic acid bacteria, so like lactobacillus rhamnosus, for instance, that already have a history of consumption from the food supply. But as you start to move away from these, you know, traditional probiotics into more what they call the next generation probiotics, so like the Akkermansia that are commensal bacteria, you know, the threshold of evidence for safety is going to change. And for the clinical study part, actually, when Paul shared his table with me before the workshop, we kind of had this discussion because his table did show that probiotics did all have clinicals in those GRAS notices. But then I started thinking, and I haven't gone back into all of those 100+ GRAS notices. But for probiotic GRAS notices, even if they did have clinical studies, they were more efficacy studies, is my what I remember.
This is Jen from Spherix, and yes, they're all looking at efficacy endpoints. And when you submit those to FDA, depending on your reviewer, they get very unhappy about probiotics having efficacy, because then that gets very close to drug language, which was also brought up by the panel earlier today. So I know when we work on probiotic GRAS determinations now, we're very careful with our language because, yeah, they're looking at efficacy endpoints. And FDA does not want your food to be efficacious.
We also can't call them probiotics.
No, thank you for saying that.
We call them live microbial strain.
Yes, we can no longer call them probiotics, even though like that's what everyone refers them to.
Okay, so you may have heard at the start of this session that we have 5 topics to discuss. We've gotten sort of through one, but that was by intent. I think this question is so foundational to establish the stage for why we're talking about this in the first place, that it was smart. It was smart to thank you for spending that much time. So it is about break time. I think it would be a good opportunity for us to refresh and come back, and we're going to move forward with the rest of the questions as if we have consensus that they're needed. So I think it's really important to look at these clinical endpoint investigations, see what we can come up with for the rest of the session. And we're going to leave these bracketed areas here. If you want to give me more brackets to consider on the break, come do so. I think that's very cool. But otherwise, this has been a very productive conversation. Let's take a 15.
I just want to make a conclusion that we currently lack validated framework that link early biological modulation in infant to durable, clinically meaningful health outcome. I think this is defining the gap.
Could you say that one more time?
We currently lack validated framework that link early biological modulation in infant to durable, clinically meaningful health outcome.
Can you give any examples of that?
Yeah, like we don't know what is the link between something we like discover early on the long-term effect on the immune system, on the brain development, on the microbiome development. So we need to decide on what measure we should be taking and what is the link of those endpoint to long-term clinical effect.
Okay. I think we're going to touch on some of that in these upcoming threads, but let's, and thank you for your comments, Mona. Let's keep this conversation moving forward. We're going to move along with the assumption that we do need to investigate additional endpoints for clinical trials. So let's start first and foremost with this question of what's the unit of evaluation? So we know the current GMS is a finished formulation where you're investigating novel ingredients or a novel ingredient in an infant formula. But is that the only and most appropriate unit to investigate the safety of novel ingredients? In other words, could you investigate the ingredients alone? This is a very broad, outstanding question, right? Is there a model, and this would be a great question for you to start off with, Susan, is there a model by which an infant clinical trial can investigate an individual ingredient or potentially a mixture of ingredients that are outside of the finished formulation of an infant formula? And if so, how valuable would that be in this discussion? You want to start that one off, Susan?
Sure thing.
Okay, thank you.
So I think the short answer to this question is that we do need to look at the ingredient within the context of the completed product, meaning the finished formula. Could we look at it in isolation? Sure. But I guess the question I ask myself is, aren't you going to look at growth anyway? If the intention is to use it in a formula, then I think you just start with a formula. From a perspective of time and money, evaluating in isolation, you're just going to spend a year or two doing a trial that doesn't get you anything that you can bring onto the market. And I think we heard yesterday, you know, obviously, if a sponsor is wanting to add this new ingredient, they have a really robust preclinical data pack. They have robust evidence from the literature and so on, and ideally understand a lot about the mechanism of action. So, you know, I'm certainly open to if somebody has an example of where this would be benefit, but generally speaking, I think that if you've got all of that in hand, why wouldn't you do the GMS?
And I guess I would push back a little bit. Same comment I made yesterday when we were talking about this, that, you know, there's not just one company involved. Obviously, this is a supplier or supply chain and a finished formulation issue. So the GMS is obviously required for a new formula, but it is not required for an individual ingredient. And so for an individual ingredient who's investigating safety of their ingredient through the GRAS or food additives program, if they choose to do a clinical, will that always be hand in hand with an infant formula company interested in pursuing that ingredient incorporation? So they run a GMS with potential secondary outcomes. Or do we envision a world in which an ingredient manufacturer themselves conduct clinical trials to investigate the safety of their ingredient outside the standard GMS because they want to explore safety questions in and of itself, right, in the context of, or in,
I think to your point, if the manufacturer wants to establish a profile for that ingredient, I take your point. But ultimately, if someone is purchasing that ingredient to add to their formula, they still need to start from square one, is how I think about this. And I also think a lot, I mean, we look today and we see, you know, how delicate a space we walk in. You know, we think the reason we spend so much time on these questions is we are dealing with the most vulnerable population we can think of. So, you know, this isn't, not only is it in adults or like we are absolutely at the beginnings of life. And I look at all the things that I read in the news. A manufacturer or one little ingredient, one little anything changes and everything changes. So I don't put much false confidence, if you will, into doing a study here and then saying it applies further down the road. That's just me, though.
I'm going to push this to our online collaborators. And actually, Dr. Ryan has joined us, Alan Ryan. I'd love to hear from your perspective. I know you've worked on in the past looking at ingredient investigations, DHA and ARA also. What do you see would be any value, or do you see that there would be any value in this individual ingredient investigation in isolation versus, you know, always within the context or the matrix of an infant formula?
For example, for DHA, could you hear me?
Yes, we can hear you.
Martech was always interested in other indications in addition to infant formula. So they were always looking at other ways to test DHA in other animals or even in humans. Also, there's a lot of studies in maternal supplementation of DHA and its effect on infant development. So there you need to look at relationship of how much you would provide a pregnant woman and how long that supplement is offered. So all these things are reasonable studies to do that may also help you get a handle on use in infant formula.
Barbara?
So there's an odd
There we go. So there's an odd aspect to this study, or to this question, the way it's posed, because if you're studying a functional ingredient in isolation, it's not in isolation. You study DHA in some sort of context because you have to have some context. And I mean, to me, the in isolation, whatever the context is for that in isolation, or the formula matrix, they're addressing two different things. And I, in my own mind, you know, yes, if I were buying an ingredient, I would have expected the manufacturer of the ingredient perhaps to have done due diligence, that they've gone through the GRAS process. And now, before I put it on the market, I'm going to test that ingredient in my formulation to make sure it's not going to cause any harm. So I don't regard them as either or, but how do they work together and when do you need both? And even when you're looking at the isolated ingredient, what else is in that formulation?
Right. Leanne? Oh, John.
Yeah, hi. Well, I've been involved in clinical trials of three types, right? I've been putting ingredients in formula, using and the DHA work and MFGM work reflects that. I've also been involved in providing syrup with zinc and copper to babies in Peru, and that's an example of isolation. The last thing, and as Alan has talked about, we have done work giving pregnant moms DHA and looking at the effects of that. I think Susan's exactly right. If you mean to put this in formula, it's probably best to evaluate it in the formula context. Although, I guess I would wonder whether or not I would ask first whether or not there's a reasonable suspicion of any interactions with existing components in formula to begin with. That might be something you do. The one advantage I would say is in looking at isolation is more of a logistic thing, and that's you don't need to, and we don't do a lot of formula feeding research right now with babies in the United States because there's such a high rate of breastfeeding, and formula feeders are in fact a very different population, and so it makes it real difficult. The one thing about isolation studies is that you don't have to have formula feeders if you provide the ingredient outside of formulas, but that may not be pertinent to the question, but it is an important logistical point. I'll be quiet now.
No, so you bring up our question 3, actually, which is, could there be value in these isolated ingredient investigations if we, were able to potentially randomize the types of feeding? That was one of the challenges that our panelists were discussing in our pre-workshop survey, is the inability to randomize the human breastfed arm. I mean, we all know this by study design, but is there a model by which we could envision individual ingredient investigation, irrelevant of or allowing randomization of the feeding type? And I guess that kind of goes back to sensitivity to the assays that we're looking at, right? If these ingredients aren't expected to exert a significant impact, would this model that we are, do you have something to share? Would this model, essentially hide the clinical effect that we're looking for if we were randomizing the types of feeding, right? If the functional effect is not strongly significant. So I'm asking a whole bunch of questions here, but kind of going to John's question of the value of individual ingredient or isolated ingredient investigations, adding it as a supplement.
Vanessa, maybe before the question I would like to ask is sort of the practical aspect. Huh? So how would you envision such studies with a background in feeding? I can imagine it for sort of probiotic drops or supplements that you would add while formula feeding, but how to see this for an intrinsic component of a formula? How would you do it?
We do this from Nestle. Like the only example I can think of is with a probiotic. So before putting it in the infant formula, we have studied probiotics on their own and randomized whether they got the placebo or different doses of the probiotic, including breastfed infants. Mm-hmm. So just as you were saying, I can think of 2 situations where we did that. It did then go into an infant formula growth and safety study, mm-hmm. but prior to get an idea of, it's more efficacy though, but study a high dose and a low dose for safety and some efficacy hints, but in breastfed or formula fed.
Okay.
Just as an example.
Yeah, thank you. Any other examples that we can hear from our panelists or the audience of where this type of clinical trial design would be? Mona.
I just want to say that using an ingredient as a supplement in Canada, we don't have a way for this. We don't have a regulation for this. It is not a standard procedures in Canada. But I will just let you know how we deal with this in Canada. Like, for example, DHA and the ARA, Martech company submitted novel food submission, and it was like preclinical and adult population. Then we decided, okay, if it is safe and it is not novel, we move to new ingredient in infant formula, where we collect literature from anywhere, that study conducted on infant, young children, preclinical, any data that can support the safety of this ingredient in the infant or very young children. If it is safe, then the ingredient, the company that have the new ingredient join with an infant formula company and submit pre-market notification for an infant formula. And I just want also to highlight the fact that here in Canada, it might be the same as the US, it is preferable to add the new ingredient to a marketed infant formula that was actually assessed and proven to be safe and efficacious. Otherwise, the clinical study design will require 3R, one with a new ingredient, one infant formula with new ingredient, one without the new ingredient versus a commercial infant formula.
Mhm. I guess I would also, to talk about Tim's point on practicality or feasibility, this still goes back to this, who is responsible for these? So if this is an ingredient manufacturer who has not done what Mona just said, is not to join forces with the large infant formula company, and therefore the onus of that clinical trial or the financial responsibility of the clinical trial is on the ingredient company, and also considering that many novel ingredients come out of startups that are VC funded with minimal funding capabilities for clinical trial design. So within those kind of frameworks, can we see, again, just thinking on the practical side, and Susan, I'll push this back to you, whereas of course ultimately a GMS will be needed and these ingredients will be in a GMS, thinking about the ingredient side and the ingredient manufacturing side, can you see a way that this would be valuable for an ingredient manufacturer to run a clinical, using some type of supplement with randomization for feeding or not? We could discuss that too, but.
Sure. So a couple of different thoughts on that, and also taking from some of the examples that are given. I think if you're the ingredient manufacturer, you're going to need to do something. So, you are not now putting that in formula, so you've got to evaluate safety of that particular ingredient as you have made it. I also think, and I've seen this a little bit in some of the questions about, a VC funded, I think there were comments from Nestle, I believe, about, doing the study first. I think that's wonderful that they did that study first so that they were able to get some additional data before moving forward with the formulation. But in the reality, that actually costs a lot more money because they had to do 2 studies instead of 1 study. And so if you tie in that VC component of your question about what do you do if you're VC funded and you have less money, the answer doesn't change from my perspective. Is that you've got to do that 1 critical study regardless, and your back is up against the wall from a monetary and a time perspective anyway. Why would you spend it on something that doesn't get you a product to the market? And then if you have an opportunity to do that, it's terrific. But I think the reality is a lot of companies don't have budgets to do 2 studies when 1 will do.
And maybe another challenge here is the fact that sometimes these innovations are supposed to replace something in a formula, alpha-lac, other whey proteins, casein innovations. So they're supposed to take out replacing something of a standard formula, and a new innovation is an add-on on top of. So how to design such a study in a pure ingredients focus, I would see impossible. You need a formula, you need a matrix.
The current regulatory landscape doesn't allow for it, right? To
Allow
test
for what?
a supplement, if you will, then as a single ingredient, and then go from that trial to market.
That actually, so the point you bring up is one that I would pose back to our regulatory folks, right? If indeed, and maybe for Nestle too, how would you address that? If you are choosing to investigate for an ingredient manufacturer, the safety of your ingredient in infants, and you choose to not do a GMS, not partner with an infant formula company, but do this kind of individual isolated ingredient, would be the equivalent of a supplement. How then can you apply the information you gain out of that clinical for the use in infant, for the use of that ingredient in infant formula, right? Is that information or evidence generation actually supportive of the use of that ingredient in an infant formula? Or does the very fact that you've isolated the ingredient for investigation, like you've just negated its applicability for within the context or the matrix of an infant formula? Am I speaking too many words?
I think that it can be useful to study the isolated component for assessing safety. Like, for example, let's say you wanted to study the effect of something that, the FDA is seeking like clinical evidence for, and then you study that in a population that maybe is like older than an infant, you could study that in that form, and that could help provide some safety evidence. Like, yeah, I mean, maybe it's not the final, like, ultimate evidence that you would need for studying for looking at the effect on an infant, but at least it would help you get towards safety.
Just
for
going back
example.
to totality of evidence. Yeah.
I'll just say from my own experience, when you are, I have a recent interaction with FDA where I was updating the literature for, I think, DHA in preterm infant formula. And the only papers that had been published were ones that were using it in a human milk fortifier. So it's not exactly a supplement, but definitely not in the matrix of infant formula. And I did my little due diligence, updated the literature, and I got a question. FDA asked, why does the use of DHA in human milk fortifier support the safety of DHA in infant formula? And thankfully, I could rely on all the other papers that showed that DHA was just fine in infant formula, and there's no matrix effect that we really need to worry about, and I tap danced my way out of it. But I think that if you are, it's a major concern. I think you can use it to support safety, but it's not enough alone because the matrix effect is important. And I've been getting more feedback from FDA when I am summarizing the literature to support the safe use of something in infant formula. They're asking for us to describe the matrix. So what is the protein source, for example? Because that's an important part of the intended use of your infant formula ingredient in your infant formula. Is it cow's milk based? Is it goat milk? Because Tony's not here, but he mentioned that yesterday. Or is it soy based? All of these things are important. So I think that it depends. You need to describe the matrix that you're testing it in and how it relates. And if you can provide a justification as to why what you tested is directly related to its use in infant formula, that's cool. But I think FDA would probably prefer it to be in the intended infant formula.
Doug?
Yeah, we were just talking about this question a few minutes ago, and so I just want to ask it again. So do the current guidelines, regulatory guidelines, allow an ingredient manufacturer to test their ingredient by taking it and adding it to a commercially available infant formula without the permission of that company's manufacturer, and then use that as an application for use in infant formulas? Can you do that?
Don't look at me. Let's see from the people in.
I'm curious.
Barbara, do you know?
I wouldn't speak for FDA currently.
Can you please repeat your question?
Yeah, it doesn't sound logical to me.
No, I agree. I think, you should couple with a manufacturer and put it in their formula and test it in their formula, but maybe that's not always possible. And so can you take the ingredient, you're a manufacturer of an ingredient, you want to test your ingredient in an infant formula, and so you're going to take and go pull that formula off the shelf that's commercially available, add it to the formula, and give it to those infants for an infant trial.
And so in that case, it seems like you're preparing something for a GRAS submission. I don't know if that would be acceptable you're not preparing something for an infant formula notification because you're not going to go to market with that product. So it seems you're more trying to hit the safety side of it.
Yeah, because you'd eventually have to have a formula manufacturer partner. Right. Yeah.
And I just want to, Jen Simmons from Spherix, I just want to chime in. If you are in this hypothetical situation, you want to use this to support the GRAS notice for your infant formula ingredient, you need to publish that. So if you're going to publish that, you probably will have to say in your methods the source of the infant formula. I know because I've been including these in all of my summaries of the published literature. And there are plenty of papers that don't say where the infant formula comes from. But if you dig around and look at what was registered in like clinicaltrials.gov, you can eventually find it. So that would be up to I'd say you probably need to get a relationship of some kind, because if you want to use it for GRAS, you got to publish it. And if you're going to publish it, somebody's going to ask.
The studies do exist, but it's a sort of in a situation where they're adding probiotic drops or these vitamin supplements on top of, and that's a different situation of selling an ingredient for a intrinsic
Yeah.
part of a formula.
Just because this came up recently, like there's no rule that you can't publish some sort of analysis of a product like a formula that's commercial without the backing of said company. So as soon as something becomes commercially available, anybody can do studies on it without support of the company. Yeah. Mhm.
So we have a Slido question, and I'll let you keep talking, Paul. That's okay. But we can talk over the question. This one is a little bit limited in the choices you have available. So we'll see what the general consensus is from our community. But Paul, you good?
Yeah, just one more challenge in terms of this hypothetical where you have someone who's like buying product off a shelf and then mixing in an ingredient. I doubt an IRB would approve that. Like there would be significant micro concerns, I think, that an IRB should have about, hey, I'm just going to mix some ingredient in here and then give it back to people to make their own formula.
Yeah, and on the practical side of things, coming from the supply chain world, we know that manufacturing process could potentially change the identity of the ingredient, the stability of it. So it really would be difficult to make draw conclusions from a study like that. Again, going to totality of evidence, it can support, but standalone is not sufficient.
I mean, and in yesterday's discussion, didn't we hear that for a GRAS notification, they want each compound individually, whereas for an infant formula notification, they want to understand the total infant formula context. So, I mean, that's carrying over from the discussion yesterday.
Yeah. Dave.
The other thing I was going to say is like what you brought up, like, for example, if you had an ingredient that you were giving to an infant who was otherwise human milk fed, I think that it could be useful for evaluating safety. And then, it comes down to basically like whether the IRB of that particular institution will say that, the potential benefit is, worth, like the risks and the benefits are, supported basically. And so, but that's all like from a specific institution's ethical board that has to make that choice.
Sure.
Yeah.
Okay, pretty clear consensus on this one. And RCT is recommended for using ingredient investigations. This has been a really cool investigation or talk about isolated ingredient investigations. Can I not move forward from the slide? I'm stuck. So we were trying to align on these, and I think we actually aligned pretty well. So conditions under which a standalone ingredient trial would or would not provide adequate evidence for safety and benefit. From what I hear, it can be supportive but not sufficient if you do an isolated ingredient investigation for safety investigations for the purposes of including that ingredient in an infant formula due to potential matrix effects. That's what I hear. Maybe you can challenge me on that. It's supportive but not sufficient, standalone, right? Supportive but not sufficient. Yeah.
So question for the toxicologist people. So for the rat studies, are those, do you use the compound spiked into the standard infant formula, or do you use the isolated compound?
Do you want to go first, Mickey? It depends. So the first thing you would do is you would do like a 90-day rat toxicology study, and you either, depending on the ingredient, you either mix it into the chow in adults, or you do like oral gavage. You can do rat studies where you mix it into like rat milk replacer. Are any of those OECD protocols? I don't think so.
So it's not.
So I don't think so. There are you can do like other studies where they have like pup in a cup kind of situations where you can isolate out the pups and feed them, but they're not like we would not include that in a traditional toxicology battery as like a first glance. Maybe I know that for like the ARA and DHA studies in rats, which is a whole nother can of worms because it's a lipid. There's only so much lipid you can give to rats before they start to get sick. But then you would give it to the pregnant dams, and so then it would be in their milk, and then they would be giving it to. So that's another way you can do it. Anything else you guys want to
Yeah,
add?
I was going to quick add just to strengthen the limitation, right? To ensure that the rodents getting the dosage that you're, saying up to this dosage, that's where the gavage, outside of chow is needed. But then you miss the matrix effects.
Yes.
So, but then it kind of goes to a piglet study where that can also test some of those matrix effects, because generally I think in piglet studies they give it in the
sow milk
formula
replacer,
kind of
yeah.
matrix. But
It's still not the formula.
but yeah, and so so I think it depends on what's the true driver of the study. Is it for safety for the ingredient? In a dossier for regulatory purpose, it's better for isolated ingredient, and then for formula kind of purposes down the road, better in a formula.
So what free advice do we give startup companies for evidence to include in a safety data package? From what I hear, if you are concerned about clinical outcomes and you are considering a clinical investigation, partner with an infant formula company and run a growth monitoring study with secondary outcomes, which we'll talk about. Agree?
Agree.
Okay.
That's amazing.
That was easy. I got that. Okay, let's move into the when. Like, when does biology trigger evaluation? This isn't following yesterday's preclinical last kind of question we asked. When should this investigation occur? And really following what we just said, even if you're not necessarily concerned that there's any safety triggers in your preclinical, when might you want to run secondary outcomes in your growth monitoring study? So we're trying to align on scientific principles that should govern this. And if you've heard me in the last 2 days, I love this tiered approach, putting some structure behind or decision tree behind when an ingredient manufacturer or a formula company, but likely we're on the kind of former side of things, when evidence would warrant their investigation in a clinical trial for physiologically active ingredients, the threshold of evidence that should essentially be investigated for determinations of needing to move into a clinical, and then the practicality of this and usefulness. So that's what we're talking about here. The first question is going back to IOM frameworks. So we do see that there already exists a framework, a tiered structure for investigating novel ingredients that have putative or known physiological outcomes or actions inside the infant. But we also heard yesterday that there's not a lot of substance in the detail when you do trigger a tier 2 investigation. So when tier 1 kind of moves into tier 2, what do those outcomes are? What models are you looking at? Preclinical, NAMs, or clinical investigations? And even though this language is not exactly correct, so where, I guess within, if I don't know if our panelists are that familiar with the IOM framework, this really was a session 2 question, but considering that framework, like what of those particular investigations do we feel a clinical is more sufficient than a preclinical model? And we talked about that a little bit already, like neurocognition potentially, right? What about immunological endpoints? This has been brought up quite a bit, and we know that some people are really curious about this. Like, which model preclinically would be great and sufficient, or is there no completely sufficient? And we do want to say a secondary endpoint investigation in a human clinical would be better. Probably in all cases, it's better. I don't know, for session 2, folks, Doug, did y'all get to the determination of an appropriate clinical model, preclinical model for immunological investigations or immunomodulatory ingredients? I think that was the survey question too, or for our session 2 panelists.
No, I don't think we did. In the survey question, sorry about that.
Yeah.
I mean, I think it's possible to do it, but I don't think specifically we got to that point in the survey questions.
Mm-hmm.
Well, in the panel, Sharon pointed out that the pigs had a more similar immune system than a mouse or a rat. So that was one thing.
Yeah, the piglet as a model for immunological outcomes.
This is Ross with Elena. I was going to quick say to add, I mean, and that's where, potentially, and this is something that is maybe a next step, where on some of those more specific developmental outcomes like immune or neurocognitive or gut, that there's sort of a more sharpened panel with those expertise to define what exactly would be the right model or endpoint. But from my understanding, when we did this assessment for our immunotox endpoint, the rat is quite validated, where if you look at, a couple angles, the TDAR is a T cell dependent immune antibody response, and then the immunophenotyping that, one of the consultants we had, Norb Kaminsky, said something around 95% chance if you do both of those and you don't see an signal of immune toxicity, that you have a 95% chance that you would catch it. So it's a pretty sensitive and specific, those are specific and sensitive endpoints to test for immune toxicity in the rodent. Not to say that a piglet couldn't be added in for that additional certainty, but at least that was just a point that I think could be explored further to define.
Well, let's add to that a little bit. Let's say that for practical purposes, an ingredient manufacturer with limited funds is wanting to do endpoint investigations for immunological activity or immunomodulatory activity. A rodent is cheaper and faster than a piglet study, and you can get some significant endpoints. Would it be reasonable and or feasible to move from that study into a partnered growth monitoring study with an infant formula company and add in secondary endpoints for capturing the same sorts of outcomes, instead of potentially going into a piglet, which also is costly and a bit more timely?
So I think we had some discussion over coffee too, that the models that we have, like the ones Ross are describing, as well as the standard rat subchronic studies, are, I think there's good evidence, and I would say consensus within the toxicology committee, that those are sufficient for establishing reasonable certainty of no harm. So do you even need to go beyond that? Maybe in some cases, but probably not much. And if you do go beyond it, is clinical the right place to go? I don't know. The other thing to keep in mind is that grass is a point in time. So you can reach reasonable certainty based on these well-accepted models, which I'd also mention, if we are coming out of this meeting saying the models that we have are not sufficient for establishing it, we got to tell pharma, we got to tell agrochemical, we got to tell industrial chemical that they got to change everything they're doing. Meaning there's good acceptance that these preclinical models can establish reasonable certainty. But let's say you do run a clinical later. Like if you run a clinical later to look at something and you do find an adverse effect, you do need to take that into consideration. That may change your conclusion of safety. Grass is a point in time. I think we heard that during some of the presentations yesterday. You're always evaluating new information. So can you reach reasonable certainty with just preclinical models? I think you can. But that doesn't mean you don't have to continue to reevaluate that as new information comes to light.
So that was our question one, Paul. So yes, I think we're continuing our discussion, maybe with the assumption that safety can be determined potentially through preclinical models, through toxicological evaluations, et cetera. And still, we are going to talk about additional endpoints that we can use. Yeah, maybe after safety is established for the ingredients, or they wanted to bring in further evidence to bolster that support, and they had a partnership with an infant formula company. Now we're talking about, can you extend that safety package by looking at these endpoints in an infant and in GMS as a secondary investigation?
So first of all, I thought the IOM 2004 framework was for more than nutrients.
It is that was,
yeah, okay.
Made it cross that word off.
And I just come back to the question I think I've asked all along, that is there some aspect of the physiological activity that was measured that says there's a need to investigate additional safety issues? What did you learn going through this? Because if you're going to go from tier 1 to tier 2, to me, the rationale is, oh, there's additional safety parameters we should be investigating. So we have to go into the tier 2 side of it. But I may be misinterpreting the question.
Yeah, because it seems like if there was a safety issue, it wouldn't progress. And so then that's where I struggle now, because then we get into suitability and benefit. So then my whole frame of thinking starts to shift, because now it's a secondary endpoint for the GMS study. So it might only be in a subset of the participants, for example. And so then I might choose an endpoint to assess immune function or gut health or neurocognition that has, less accuracy, less precision. So, how much emphasis do you want to place on that anyway?
Yeah, so we're not out of question one yet, is what we're saying.
We're not.
This is a challenge, and our partner from Nestle, would you like to add some comments as to why this is still a valuable conversation? Like, I guess the point is that companies are doing these, and one of the reasons that we're here in this room is that there is some challenge for certain ingredients to even make it through the GRAS notification program due to concerns, right, that are difficult for us to suss out, but we're trying to answer them as best as possible. And if we are not able to articulate it clearly what that safety concern is, which it does not seem like we've gotten there yet, like where can we go with this conversation if we can't fully say we're answering this question on safety with these endpoints?
And I'm not saying that there's one specific safety area that we should identify. It's more a matter of, does the physiological activity raise additional safety questions? And so that depends on the compound that we're talking about.
Yeah, in a quick case in point with lactoferrin, I think although there's been 100+ studies and lots of in vitro preclinical studies that highlight some functionality of the immune system, I think, although never has there been adverse effects to the immune system, I think just all the totality of the evidence says, hey, there's some immune functionality here that then justifies, further look into this safety aspects of the immune system. So that was generally, I think, FDA's recommendation as it related to lactoferrin. Although evidence wasn't there of an adverse effect, there were physiological effects related to the immune system that then justified looking at it closer.
Maybe can you tell us more about, like, because we're looking at questions about like what kind of endpoints could be valuable for these kind of studies. So do you want to tell us about like what kind of endpoints you used?
Well, I would say, so from an ingredient point of view, I would argue that the types of the preclinical studies broadly for most ingredients are sufficient. And that's where then if there are immune questions for certain ingredients, I think the Callahan paper that was published a couple of years ago, I think highlights a pretty good laundry list of endpoints to look at. Whether that's needed for every ingredient, I think that's then on the company to determine. For our ingredient, these are the most important endpoints because of what we know in the literature about the ingredient's functionality. So it's not necessarily that they got to do everything that Callahan lists, but those endpoints that are relevant to their ingredient. But Jody,
I made a comment on the break that you may be referring to it. And I think it's like, I don't want to add a lot of complexity and say we have to do all this, but I was saying as an employee of an infant formula company, like we're adding them because we think they are going to bring a benefit. And if there were things we could do to make parents feel better about safety, we would be willing to do them. So we're doing the growth and safety studies anyway. We never have done one that only was growth that I can think of in 25 years that I've worked for the company. There always are secondary endpoints because we want to show, it's a lot of effort to show that there is a benefit. So of course we want to show a benefit. And if there is a safety concern, we also want to make people, ourselves and other people, feel better that it's safe. So I think it's like, we don't want to add complexity and say everything in that paper you have to do, but I am hoping to get some practical things if there was a signal, because to me, we're looking for safety signals. But if we want to reassure safety, which is somewhere between a requirement and what you want to do to feel better about it. So it's not that I'm saying I want a full list, but I am interested if there is something we feel needs reassurance, not a list of 12, but what would be the minimum thing we would go after? And, we have a list, but I feel like people also want to narrow it a bit. Not ready to make it mandatory, but I am interested in what experts think about how to reassure safety, maybe is another way of looking at it.
Yeah.
So Jody, I'm just wondering, so from your past experience, what of those secondary outcomes that you have measured in the past could be thought of in the frame of a safety perspective? Like,
Well, I mean,
yeah, can you
the
give us some
types of
examples?
ingredients we're adding are getting more sophisticated too. So I think suitability could be softer stools. Like when I say there's a minimum, or it could be a microbiome, or it could be a marker of gut barrier, or it could be a marker of immunity in the gut, like an SIGA. But I think those are benefit. They're not, things I've heard about reassurance of safety are like IgE, IgG or so, something where we could say they're not reacting to a new protein, things like that. I think we have a good idea of ways to think about showing the benefit, all the markers in the gut. I think we're getting more sophisticated about what we can look in the blood in terms of an effect. I'm just not confident in what reassurance of safety would look like. And even when we get all these immune markers in the blood, even if you look at a breastfed reference, to be honest, I don't feel like we really fully understand what's happening with the breastfed infant because we don't have these serial measurements. But that's a whole nother topic. So
I might even suggest maybe you're already doing it. So if you're adding immune markers to a growth monitoring study to look at effects, I think if we go back to, are those markers getting closer to human milk? Are they getting further away from human milk and not the same as your other formula, your formula without the ingredient? Like, I think that can be the proxy for safety. Like, as long as you're not getting further away from human milk, like then you're adding appropriate markers for immune function based on your ingredient, which is going to be case dependent. Like, hey, we think it might impact this element of immune function. Okay, well, let's look at that. If it gets closer to human milk, not a concern. If it stays the same, not a concern. If it gets further from human milk and not the same as the other formula, then that's a concern.
Yeah, it sounds like a lot of us want to talk about what the specific endpoints are that we could be investigating. And for this question in particular, this was more a threshold of evidence. When would the evidence trigger or warrant an RCT or a secondary endpoint in a GMS? And I think that I'm hearing is we don't really have a clear one. I mean, safety is established in preclinical toxicology studies, et cetera, as we've discussed. There might not necessarily be a regulatory or, legal precedent for saying you need to then put that into an infant clinical, but many are already doing it. So let's kind of say, if you are already going to be putting that ingredient in an infant clinical, let's now discuss what the potential outcomes are that you can gather. Yeah, well, let's wrap this one up first. This is the threshold of evidence question. At what point should demonstrated physiological activity in a functional ingredient trigger a higher tier of clinical evaluation? Yeah, if you look at the response options, we are kind of throwing in there benefit in addition to safety. Yeah, so we'll kind of see. Well, we'll see what you guys answer. Yep. Hmm. Okay. I'm kind of surprised with these results, actually. I would assume it would be more case by case based on what we're talking about, but it looks overwhelmingly like if there is a trigger. But again, I would challenge all of you who are answering that. If there is a trigger that is a health consideration or a safety consideration in a preclinical model, wouldn't that stop your investigation?
It totally would stop the investigation.
Yeah. Like, how can you justify taking something with a safety trigger in an animal model and say, okay, let's put it into infants?
Mm-hmm.
This is.
I
22
when I
hours.
was answering that question, I was thinking more about like if there was something in literature that suggested like a function of a thing, not like in the toxicological
Not a negative
study.
safety trigger.
Yeah.
Okay.
Really quick.
Point out one of the options is only if there's a proposed health plan.
Is your mic on?
Yeah. Oh, there. Is that better?
No, we might be out of battery.
Oh, maybe it died. Oh, no, there it goes.
It's good.
I'll yell. So only if there's a proposed health claim. Technically, you can't have a health claim on infant formula because health claims are about reducing your risk of chronic disease. And the kinds of claims that would show up on infant formula are structure function claims. It's about structure and function of the body. So just terminology becomes important. And as this rolls into a report, I want to make sure we're in the right place.
I also just want to, hi, this is Jen from Spherics. Just because you mentioned literature, I know that when I've had pre-notification meetings with FDA for GRAS ingredients, they're searching the literature. And so even if the literature is reporting some like immune function in a in vitro model, they may ask you about it, or they may ask why your current data like does or does not support a role in the immune system. So, you know, do your Googles, guys. And when you're writing up your publications or your GRAS determinations, this may be something that FDA may ask. They may ask, you know, why this publication in these other models, why is this not a safety concern? And I think like, yeah, with lactoferrin, for sure, there's so many things in the literature. I think that they tried to look at lactoferrin as a treatment for COVID.
Mm-hmm.
And so there's a lot in the literature.
I'm just kidding.
No.
Yes, there's a lot in the literature.
And yeah, like, and FDA, they can use PubMed just like we
Exactly.
can. So
Yep, exactly.
if it's in the literature, it's something that you may need to consider.
Yeah. Okay, let's move on to thread 4 in the interest of time, which is really our bread and butter. This is endpoints, right? What can we actually measure? And in this conversation, we will try to take into consideration practicality and feasibility, right? So yes, can we collect babies from all infants? Not likely. And therefore, what can we actually measure from these GMS studies? What endpoints can realistically evaluate whether functional ingredients, physiological activity is appropriate for a developing infant? We don't have to go into the appropriate for development, but we could, again, circle back to safety. If at all possible, it'd be cool if we had a short list that we could develop here today, but I'm not sure if that is possible. We'll see. And the conditions under which decentralized and home-based trials might be beneficial for generating scientific data. Yeah, okay. So let's start with that. Just looking at these endpoints, and maybe we can start with that. I mean, do we have that second question up here already? The second one. So non-invasive tools. I think it would be nice to start with that second question because we do want to push for as much inclusivity as possible. Like, can we apply these to as many trials as we can by not doing invasive measures, right? So yeah, Susan and Leanne, both of you have worked with decentralized trials and the benefits of those. And you speak to some of the valuable non-invasive alternatives that can collect endpoints that would be interesting to measure.
Sure. Do you want to start? All right. All right, we'll put the ball right there.
Yeah,
Okay, very good. So in short, we do have a lot of experience with doing growth monitoring studies, and particularly over the last 4 years, considerable experience with doing decentralized clinical trials for growth monitoring, meaning fully decentralized. And almost every study that we have done has incorporated one or more of the non-invasive alternatives that are listed on this slide. So I can tell you that I have practical experience from beginning to end with collecting stool, saliva, urine, buccal, and behavioral data, all from the caregiver and baby's comforts of their own home, not only reliably, but with what we believe is a lot better data quality than we had previously been able to collect. We are seeing incredibly high compliance rates for our caregivers to submit these samples. essentially everything on that list is coming in between 95 and 99% sample collection. So in terms of practicality, feasibility, we ship everything direct to the caregivers. Depending on what it is that's being tested, obviously they're trained, and sometimes if it's something very simple, we may offer a digital training video with a how to do or how to use or how to take a stool sample type of thing. If it's something kind of in the middle, we also do a lot of televisits where we have our study coordinators who are nurses in pediatric or neonatal units who are used to working with infants that will get on telehealth calls and walk a parent through these types of things. And then also, I think, to John's work earlier, there are not all, but there are a lot of cognitive assessments that can be done remotely. So there's opportunities to do those as well. So I will stop there because I'd be very curious to hear about your experience, Leanne.
So again, in sort of more in-house clinical trial settings and less of a remote setting, I mean, parents and researchers can be pretty adept at collecting stool, saliva, urine. The behavioral cognitive batteries are easier, but it requires a clinic visit. They're fairly long, specialized training as well to do it properly, like the Bailey ages and stages, for instance. I will say, though, blood draws aren't out of the question. I mean, we should be thinking about what information we can obtain from a dried blood spot. There are many validated assays now that you can use a dried blood spot. So parents are definitely amenable to a heel stick throughout the entire first year of life. I mean, once you come at the infant at 1 year with, a needle and tubes, and there's multiple tubes, I mean, that's a no, but I think you can get a lot from the dry blood spot. So I would add it to the list, actually. We find urine collection a little tricky. I mean, we can send home the urine collection bags, provide instructions, but there's often contamination. We actually get much better compliance with all these things if we just collect the diaper, the entire diaper. And we can provide one that's a little cheaper, so they're not as absorbent. Some are getting like a drop out of some of the new ones is quite difficult. But, we need to think about as well, sometimes in these trials, the health literacy of the people that come for these types of studies. I mean, we definitely see a difference in the parents and caregivers that would come from an NIH-funded trial that would have a very sophisticated question compared to parents that would come for a growth monitoring study. We see differences in health literacy. People are coming for the free formula. So we need to make sure that the types of secondary endpoints that we're collecting are definitely suitable for the health literacy, and people are understanding why it is we're doing what we're doing, because that's informed consent. Yeah. So we can talk about participants later. But
in Santiago, Tom Walter, looking at iron deficiency anemia, we did sleep studies, which is kind of cool. Yeah.
Yes, very cool.
So electrodes on baby's head and measure the responses during the sleep.
I think just to add, I'm sorry.
Go ahead.
I was going to say, just to add to that, we too have had success with the blood and the urine draws, but we've also done a number of studies where we're looking at some more novel endpoints, again, toward crying and fussiness, wearing the baby onesies. I think we've talked a little bit about the importance of stool consistency, and in some cases you can train caregivers to do that, but we've also have some data to suggest that it may be better to have parents take photographs of that stool and submit it, and have our central raters actually rate that. And we've published on this, showing we can get a better signal to noise ratio by having trained experts rating stool consistency. What we have seen overall is that parents tend to gravitate toward extremes, meaning like either all fluid and diarrhea, or just they actually won't tell you a baby's fully constipated. It's almost impossible to get a parent to report anything bad about their baby. So they like
They're
take
precious.
it, they do, they take it so far. So you see, and then you take those same photos and you put them in front of experts and you see a lot more of a normal distribution. So I think there's a lot of things that we can do, not only in terms of convenience, but also the quality and getting better signals out of some of these more novel endpoints. And sorry, Barbara, I didn't mean to interfere.
No, that's important, because what I wanted to add to the discussion is, FDA has a long history of using validated biomarkers. Sort of, when you look at the whole health claims, most of our health claims are based on a validated biomarker. And it sounds like you all are collecting the kind of data that could enable a validation of a biomarker as predictive of some other long-term input. And again, there's just a long history of having to use that, especially on the food side, where on the drug side, you can do the long-term studies to get the definite health outcome. But on the food side, it's much harder to get that kind of body of literature. So, and I was, they've used things from NIH to do that. I know we've looked at what the drug folks used as an endpoint or as a marker. There's a NASM report about validated biomarkers. And just another addition to this, for the 2020 Dietary Guidelines Advisory Committee, as Sharon pointed out, it was the first time that the guidelines were to begin with toddlers or begin with newborns and go all the way through. We had questions about behavior, cognitive function, and the tests were just all over the place. And in fact, we even brought in a consultant to help us try and interpret. And we were basically told we couldn't really draw strong conclusions from the nature of the literature. And to me, it was just simply an indicator that for this area to really bloom, there does need to be some consistency, validation of what do you get from that and what does it predict long term. So it sounds like you already are well on your way to producing the data.
And I'd love to hear also from John and Alan for your experience in the neurocognitive world.
Well, the people are really interested in using parent questionnaires for neurocognitive, and I would just caution everyone that the things like the ages and stages, someone just talked about signal and noise ratio. The problem with that is that you are getting what the parent thinks the child is doing, and the studies that I've seen suggest that while those parent reports might in fact be predictive to some degree of later outcomes, they don't actually correlate with what the child actually does. So what you're getting is a picture of what the parent does, which is important to some degree, but not if you're putting a nutrient in the child, you're taking a real chance in using those. Now, if you use that as sort of an in-between measure and get a real behavioral measure, I like, Alan talked about using sleep measures, which can be collected remotely, or and something like that, and that's objective. But I worry about those kinds of things. And the other thing is that the Bailey scales, especially during the first year, are not predictive at all of outcomes. And I see a lot of clinical trials sort of hang their hat on like trying to improve the Bailey by 5 points during the first year, and it's just not a meaningful outcome. And so this is a really good question, and the problem is that we don't really yet have very many remotely administrable outcomes that would actually work. And there's a bigger question when you get to question 3 that I'd like to address as well. But this is a real problem, and I worry that we're missing a lot in looking where looking for our keys where the lights are the brightest, as opposed to really digging in.
Yeah, and we can address that question 3, John, if you want to jump right into the tools that are available.
Yeah, my big problem is that there is a reliance on things on global developmental tests like the Bailey and the Mullen and all of that. Tests like that, I don't know, the Kaufman or the Griffiths in the UK. Those measures all overlap because they all stole from each other when they were developed in the 1930s and 1940s. And so they're all basically the same thing. And, they were designed to detect developmental delay, categorical. Are you delayed or not? Movement of 5 points on that scale is not really what those tests were designed to do. And yet at the same time, I see clinical trials designed to try to move that distribution when, in fact, I've seen a couple of studies come out showing that a nutrient actually reduces the degree of developmental delay, and then have that result sort of poo-pooed or de-emphasized in the literature. That's really what those tests were designed to do. The second issue that I have with this is that it's becoming clear that certain nutrients have specific effects on certain cognitive components or certain cognitive cognitive functions, and it's important to note that these cognitive functions, especially in early life, are relatively independent of one another. They're dissociable. So you can be good at this but not good at that, and a baby is sort of a mishmash of things that are mature or things that are immature, things that are emerging, things that are developed, and I worry about the reliance on the global tests because you can very easily miss something. Those global tests, like the Bailey, for example, conflate motor function with imitation, with language, and all that, and they're not the best idea. They would not be the best instrument to use if you are looking for effects in a particular domain.
What about visual acuity?
Visual acuity is great. Yeah. Would you measure visual acuity with parent report though? No. But if you use visual acuity, now it's interesting that the behavioral measures, if I remember right from DHA literature, the electrophysiological measures of visual acuity actually showed bigger effects than, for example, the behavioral measures, suggesting that the behavioral measure has a significant amount of noise in there. But visual acuity is fine. Yeah. It's not a behavioral measure. It's not so much a cognitive measure though.
Right.
Any additional thoughts from you, Alan?
No, I'm good.
Okay. Well, punching it back to our researchers here on the table and to Susan for clinical trial design, can you take the input from John and apply it to how you do your neurological or neurocognitive evaluations?
In short, absolutely. I think, and this is what I was talking about a little bit earlier when I was saying we need to put together the guidance and the standards from the experts in neurodevelopmental in terms of what are the assessments that are correct. Where these assessments are done is a detail, right? If they're done at a site or need to be done on a site on a yearly basis or whatever the appropriate cadence, those are the details. But what we really need are a good understanding of what are assessments. When our neuroscientists and clinical psychologists look at these assessments, in general, to your point, we have a lot of hammers out there in the toolbox. We are very much aware that here we are in this day and age, and we're still using these very old tools that were developed a very long time ago. So would be one of those, please don't get me started on how bad the instruments are that we use. We won't get out of here on time. But so I couldn't agree with you more. Caregiver reported outcomes have their place and they can be absolutely valuable and give you one set of data. Clinician reported outcomes similarly have their place and they can give you another or complementary set of data. I've hardly ever seen one trade for the other. Usually they're telling you similar but somewhat different things. So I'd love to have the in-depth conversation where we lay out the roadmap, because the other thing is, it's often the case that you can start with one assessment, but then you age out of that assessment as you get to the second year and the third year and the fourth year. And so it is a complex space that we can't solve here quickly, but I know that as a group, we could.
Mm-hmm.
It sounds very similar to the conversation yesterday on preclinical models and how to develop standardization for these models so that they are universally accepted as investigative platforms. So a roadmap can be developed here where we identify these are areas that we need to develop consensus on for endpoint investigations. Neurocognition sounds like one that we are talking about pretty extensively. What about immunological investigation? And actually I want to pull up, this is one example, I do want to pull up our survey, because we did ask our panelists, these and all other panelists that you've heard over the last couple sessions, about the Callahan paper and endpoints derived from the Callahan paper, and asked for our panelists to rank them. And so I will report those ones. I thought it was quite interesting. And I would have expected the vaccine response would have been a very top tier endpoint that you're looking at, but it was not. Secretary IgA levels were the top ranked response for endpoint investigations for immunological or immunomodulatory ingredients. Yeah, so we did not tease out the difference in whether we were asking about for safety or for benefit. And I don't have the question in front of me to read off.
Argued it in the thread.
Yeah, that was the discussion that was prompted by this question of, well, if we were investigating safety, it'd be one thing. If we were investigating a claim or benefit, it would be another. But let me finish the list, because I think it's still interesting. Secretary IgA is the top. Cytokine profiles as the second, lymphocyte subsets and activation markers as number 3, vaccine response titers as number 4, and then the others were below that one. But quite interesting. This ranking certainly was not differentiated based on safety versus suitability, and it also led to this question on collection, specimen collection. And most actually argued, like, although this is kind of nice to have, like, what's the realistic feasibility of collecting blood for these endpoint investigations? But we heard from Leanne, like, we shouldn't necessarily automatically dismiss the blood markers just simply due to collection feasibility. And Leanne, you've had experience in the United States. I know you're in Australia now, but your experience is running clinicals here under this this domain, right?
Right. Yeah, right.
And it's with the RBs, one set of level of experience, then with, talking to parents and caregivers, and then what is acceptable to the participant, which is the infant, and then toddler. Yeah.
Are those trials all done on site?
All of ours have been done on site with combinations of things that have been done with wearables and app data collection. We'll often send things home with parents for biospecimen collection at home. We find if we extend the window in which we allow the specimens to be collected, we're more likely to get a specimen than we're wanting it to, the child to defecate when they're at the clinic visit. Never happens.
Yeah. Unless you don't want it to happen, right?
Yeah. And Susan, do you ever incorporate blood collection for decentralized trials?
We have. That doesn't really necessarily fall on the non-invasive list anymore.
Mm-hmm.
So I think, kind of common sense is the prevalent trade wind here, in that if you want to collect stool and urine and things like that all day long, we'll send out kits, we'll get them back. You're going to get a great sample, whatever you're evaluating, it's certain. I think blood, we've also gotten great numbers with it, but we really try to ask the question, do you need it? Don't do this if it's a nice to have. Don't do this if you're just curious. Like, please go back to your clinical data pack and work out as much as you possibly can. If you need it, we can get it. But one of the things that you will, you're competing against everybody's studies for these participants. And if 9 don't have a blood stick and 1 does, you lose. So, it's not that you can't get them, but you will. We've gotten them. We've done very well getting them, but we just say, if you need it, we'll figure it out. And we have. So I think that's where we stand with all of this. And what can you get from micro amounts of blood? And then, of course, we have had studies with serial collections, really only one that, and it's tough. I mean, you're gonna take a lot longer. You're gonna have a lot more drop off. Parents don't like putting needles in babies, like to say obvious dumb stuff, but it's just the truth. But if you want to collect microbiomes, let's, game on.
Well, how, for the immunologists in the room, how valuable would be salivary secretory IgA or stool secretory IgA instead of blood IgA for looking at an IgA response? I say that because I am a secretory IgA researcher and I already know the answer, but just kidding. It is a question, like if we're going to say, non-invasive endpoint collection is best, like gold standard, then what of our immunological endpoints can we evaluate with saliva and with stool and with urine? I mean, you're really not immunological by its nature. Any thoughts on that one? Or what about fecal calprotectin? So they're nice to have, but they're not replaceable, right?
Well, or validate them for some purpose.
For some purpose.
Yeah.
Just to understand, Vanessa, are we now still talking about safety or function?
This is under the previous threads discussion that an ingredient company has validated safety with preclinical studies and still would like to enhance and bolster their safety package with a secondary endpoint in a clinical trial and maybe get some claims out of it too. I mean, right? Let's just throw it all in.
Actually, I want to bring up that's something that is kind of a jump shift from what we're talking about. And that is the fact that the dietary guidelines for Americans now do include recommendations that actually reach into pregnancy as well. And, for the standard process for the dietary guidelines is you appoint an advisory committee. They use 3 techniques: food pattern modeling, systematic reviews, and data analysis, looking at things like NHANES, to develop a set of conclusions. Sharon was key to what was done on the 2020 DGAC, where, we brought up issues like peanuts and developing allergies. There were a lot of other topics we brought up, generally with the conclusion of insufficient data or very limited data to draw a conclusion. But assuming we kind of get over the current status of dietary guidelines, it is something to consider for the future, because the kind of questions we're asking of what can you measure to be predictive, that will be looked at very carefully if, in fact, the dietary guidelines for Americans are going to have a very robust set of recommendations for infants and toddlers. So it's, and one part of it is to look at the study design and whether or not that study design is consistent with what you're trying to address. So Sharon, I don't know if you want to add anything about your experience with the DGAC.
No, I think you summed it up really well. I mean, there's just a paucity of evidence, and I like when you even, like you said, to try to look at like what are normative immune parameters in breastfed infants, because we just don't bleed healthy breastfed infants. Mm-hmm. So there's a couple studies out there, and I think that those should be utilized more. But yeah, I would say in most of these areas, we just don't. And then just the whole concept of human milk composition, I think that was brought up. So there's new evidence. But like for lactating women, we had 10 systematic review questions, and 9 out of 10 there was insufficient evidence. And the one that we could conclude was if you give women DHA or they eat fish, their milk DHA goes up. And that we know that already, right? Mm-hmm. So yeah, lots of work can be done. Yeah. And it would help this whole process. What we're saying, if we got better human milk values and we can actually do new AI, actually move beyond AIs, actually have DRIs for infants, then that would inform what our infant formulas should look like.
Right. And just to once again come back to claims, that for foods that are not designed for infants, I know the industry always looks very closely at the recommendations in the dietary guidelines to see if it's an opportunity for a FDAMA claim. And anyone who wants to know more about FDAMA claims, we can talk. But it's an authoritative voice, those recommendations in the dietary guidelines, and that then connects with other federal programs.
Okay, we're not going to make it to our last thread, but we have talked about it already, which is the portability of evidence, and I think we covered that pretty sufficiently when discussing clinical trial design and appropriate ways to investigate individual ingredients versus in the matrix of an infant formula. So I'm going to say we've reached the end of our threads, but I do have one more question to pose back to the panelists, and it's kind of following what we just heard from Sharon. Yeah, so if you could say what single scientific question or scientific infrastructure that, if answered, would most advance this field, like what would you say? Actually, let's start online. Mona, if you're still there.
Yes, I am here. Yes.
If you could identify a single scientific question or scientific infrastructure to, if answered, it could really advance our field, what would that be?
Okay, my question would be, can we define a globally accepted evidence framework that reliably distinguish biologically meaningful developmental effect from benign physiological modulation when evaluating non-nutritive functional ingredient in early life?
Okay.
Or put it another way, can we define what constitutes sufficient developmentally appropriate evidence across jurisdictions so we can reduce duplication without compromising scientific rigor?
Mm-hmm. Sounds like standards developments would be needed there, right? Reference ranges, et cetera. Mm-hmm. Alan?
Looking at the future, it's going to be hard because in the United States, most babies are breastfed. So the question is, what's left in terms of infant development for those who cannot breastfeed or have to go back to work, and the kids who have to be fed infant formula? It's necessary to provide them with the best possible formulas that can be produced. And I think right now the marketplace is being flooded by a lot of formulas that may or may not be the perfect food supplement for little kids.
Mm-hmm. So every time you say that most babies are breastfed, everybody in the room shakes their head.
Yeah. Well, at least early on.
Well,
at least in the United States. Yeah.
They leave the hospital saying that.
Yeah. Okay. Yeah. We just wanted to clarify what you meant by that.
Yeah, and we're not allowed to recruit them if they leave.
Right.
Yeah, and that would be an infrastructure improvement.
Got it. Yeah.
And John, to you then.
So think about this. Assuming that we would be able to study moms who start to supplement and at 6 or 8 weeks or whatever. I live in a world where part of my day job here is to study populations of relatively low incidence developmental disabilities, and one of the ways that the federal government has facilitated that is to establish centers that are harmonized, that can actually and work off registries and centers that are ready to run clinical trials that basically use medications or multimodal treatments for kids with disabilities. It wouldn't be a stretch for there to be developed across the United States a series of centers that were ready and willing to jump into clinical trials that would that could be harmonized, that would address the burning questions of the day, whatever those would be, and I'll leave that to you all to decide. But basically having an infrastructure of sites in different regions that would be able to carry out these clinical trials and work with one another in a harmonized way, that would be a really big infrastructural
Great, thank you.
improvement.
And for our in-person panelists, we only get 1 minute each. So Susan.
Thank you. So I agree a lot with the comments offered so far. I think that very simple things that we could do within the people that are in this room, like starting to write these things down, getting beyond, the consensus paper is step 1, but step 2 is guidelines. Client after client comes to me looking for advice, and I don't have anywhere to turn. I'm looking at a PowerPoint from the FDA from 2021 that lets off, and everything we're talking about is Brave New World. And within this group, we have preclinical experts and ingredient experts and immune experts and cognition experts, and we could lead the way and say, let's do this and let's do that, and these are needed and this is aspirational, and then set a cadence to updating these things as it go. Because right now, Doug has showed us that science has marched on as it always has, and he's way out there all by himself. But and we want to figure out, how do we close the gap? How do we do it the right way, cost effectively and with certainty and in the right time and place? But there is so much we could do today without doing another experiment that would drive the field forward and harmonize the quality that we're able to bring forward, so that the right IP gets onto the market and the wrong, whatever isn't working, doesn't get there.
Thank you. Leanne.
Yeah, framework and strategy, right? Just like the GMS trials have weight, length, and head circumference, we need to come out with a set of small measures that can be used to evaluate these different physiological activities that we think are important. Right. Then, like Sharon has said, then we can contribute to a larger body of evidence where we can then evaluate individual ingredients and inform dietary guidelines eventually, because we'll have much more data available.
Mm-hmm. And Barbara, I'll close it out with you.
So everyone said great things. I'm going to add to it. I was shocked when the Dietary Guidelines Advisory Committee was told by USDA they preferred we not use their human milk data because concerns about the quality of that data. So I know we're making progress, but I think that is key to being able to move forward, to know the composition, know the kind of variability that is associated with it, and then also think about how that relates to the DRI process, where we can make more concrete recommendations. Referencing our last discussion, I do think more effort is needed to make sure we understand what could be valid biomarkers that allow us to predict the future impact of adding some of these ingredients. It could be a very powerful tool.
Okay, wonderful. And with that, we are going to close out our session 3 and our workshop. I do want to just thank so much everyone who has participated in this workshop, everyone who came today and yesterday. This has been so much of your time and energy, and it has led to really, really fruitful discussions. So thank you for your in-person participation, for your virtual participation online, for the workshop planning committee. Holy cow, that was a lot of work, wasn't it? Yeah. And for Cypress and Stefan, who've done just a really, really remarkable job leading us to this point here today. And that's it.